Hi everyone, good morning. Good morning po.
Morning po.
Ayan.
Morning, good morning.
And good morning po.
Morning.
Good morning everyone. And kumusta po kayo? Yan, sana po okay po kayo ngayon. And currently we have 168 attendees. Ayan, participants who registered for this FishKwela webinar. Okay. So before we start, allow me first to acknowledge the presence of our guests and speakers for this event. So yung speakers po, we will be introducing them to you later. So we have here Doc Muji Santos, our Training Division Chief, Ma'am Norvi, our Training Section Chief, and Doc Janelle Laura Gacad. We have also here Dr. Jonathan Carlo Briones, Dr. Karin Chuchida, and so far, sila pa lang po ang nandito. Ayun, sige po. Mamaya po, makikilala pa natin ang ibang speakers natin. So, ayan. Welcome to our webinar on ParaMapping, Morphological and Molecular Techniques in Parasitology. The goal of this activity is to educate you participants on morphological and molecular techniques employed in detecting parasites in fish and other aquatic and semi-aquatic organisms, and will also highlight the recent advancements and findings from studies regarding genetics of various parasites. So, hindi ko na po papatagalin. Let us all put ourselves in the presence of God through the recorded invocation to be followed by the singing of the national anthem.
kababayan, ang pambansang awit ng Pilipinas. Kayang magiliw, pero sa sinakalan, alam ng puso, sa titik mo'y buhay. Upang hinirang, kuya ka ng magiting, sa manlulupin, di ka pasisigil, sa nagatang tutok sa simoy at sa langit mo bagaw, ay ginatang tulad, awit sa pagkayang minamahal, ang isang ng matawad mo'y tagumpay na nagtitindig, ang bituin at araw niya kailan pang ay di magtitindig, lumalang araw ng walatin pagsingka, buhay langit sa piling mo, ang higaya na pagkay mga amin, at mamatay ng dahil sa'yo.
Alright, good morning once again. Ayan, welcome po sa atin pong FishKwela entitled ParaMapping, Morphological and Molecular Techniques in Parasitology. So today is Wednesday, October 30, 2024, and I am Lei. I will be your host for this event. So ayan po, we have participants po, ayun no, i-acknowledge lang natin yung mga participants na lang may message sa atin pong chat box. Ayan. Meron po tayong participants from University of Rizal System Cardona, College of Fisheries. Good morning po. And we also have participants from Misamis Oriental, Bansud Oriental Mindoro. Then we also have participants from Mindanao State University. And good morning po sa mga taga-SUC. May mga taga-LGU din po tayong mga participants. Also, we have participants from national government agencies such as before. Then, yan. Sige po, please comment po from where you are from. Meron po na message from before Dagupan, Pangasinan. Ayan, sige. And Camarines Norte State College. Sige po, keep them coming. Laguna State Polytechnic University College of Fisheries. Good morning po. Then we also have from Cagayan State University Aphari Campus. Then from Camarines Norte also, ayan po, may mga participants po all over the Philippines from Luzon, Visayas, and Mindanao. So welcome po to our FishKwela on ParaMapping. Okay, so for the attendance form po, you can access the code, QR code posted on the screen and through bit.ly/attend paramap 1130. Okay, sige. Ayan. So, are everyone excited po sa atin pong event today? Pwede po bang makahingi kami ng mga reactions? Heart reacts, clap reacts, ayan, like reacts. Ayan, from Mindanao, Cabacan, Cotabato. Hi ma'am, good morning po. Ayan, meron din po from Eastern Visayas State University, Carigara Campus. Ayan. Yung link po, ayun po, yung link po sa atin po mismong slide, bit.ly/attend paramap 1130. You can also scan the QR code po para mas mabilis. Okay, sige po. And sa iba po, ayan, okay, sige po. Alright, so to formally start our opening program, let us all welcome the Training Division Chief of NFRDI, the one and only, Scientist 5. Oo, marami siyang ganap, pero he made time para po maka-attend dito sa ating event. So, let us all welcome Doc Muji Santos.
Hello, magandang umaga. Thank you, Lei. Magandang umaga sa lahat. Good morning, good morning. Ayan, dami nating participants, Lei, at kitang-kita natin yung ating mga reacts. Hi po, I'm here now in Leyte, doing some also in-person training. Ayan. So, let me see if I can do this, Lei, in different formats ng magandang umaga. At pasensyahan niyo na kung medyo magkamali ng konti. Naimbag nga aga, maya pa yabak kekongan, maayong buntag. Yan. Tapos, buenos dias, of course. And then, good morning to everyone. Pwede na ba, Lay? Pwede na. Kailangan ko pang practicein yan at pahabain pa yung aking mga iba't ibang good morning. All right. So, yeah, we are sure that everyone is excited with this ParaMapping FishKwela, regarding talks on parasites on aquatic organisms. And so, balita ko, tama ba, Lay, na sold out ang ating slots? In fact, may mga nagme-message sa akin na kung mabigyan ko daw sila ng slot. Sabi ko, sorry kasi nga hindi ako may handle ng slots. But yeah, I heard it's about more than 1,000. Tama ba, Lay, yung nag-register?
Yes, Doc. Currently sa record po is around 1,250. And
Wow. Oo nga. O, sana lahat sila makapag-online din, ano. And of course, we made sure na ito ay magkakaroon din ng CPD points, no, para dun sa ating mga fisheries professional. You'll be earning around 3 points for this one. Now, again, on behalf of our Executive Director, Dr. Lilian Garcia, and then the entire NFRDI family, welcome po dito sa training na to. This is one of our ways of fulfilling our mandate under the Fisheries Code, Republic Act 8550, in terms of training, capacity building, and sharing our experiences. This is one way of keeping our promise, so to speak, for all of you guys. Now, of course, kailangan nating papasalamatan yung ating mga speakers sa pangunguna ni Dr. Janelle Gacad Manlises. Siya po ay isang balik scientist, hosted ng NFRDI this year. So, thank you, Doc Janelle. And palakpakan din natin, of course, yung DOST Balik Scientist Program. Ayan, talaga naman pong full blast na po itong program na to. And marami pong mga Filipinos that are availing of this program. Bumabalik po sila para po makatulong sa atin. And sana nga po si Dr. Janelle Gacad na recipient ng itong program na to. So tangkilikin po natin to. And this Balik Scientist Program is, for this case, through Picard na po ng DOST. Palakpakan din natin, patenkyuhan din natin, of course, si Dr. Arvin ng UP Baguio, si Doc Karin Tsushida ng Kyoto University. So we're going international here. Si James Owen, who is a PhD student from Seoul National University, and Doc JC Briones, from University of Rizal System. So thank you very much, guys. Thank you, Dr. Janelle, for coordinating this program. I think first time ito na gagawin ng NFRDI talking about parasites sa mga aquatic organisms. So thank you very much, Dr. Janelle. We're looking forward to more great work, more collaborations, and hopefully, sa ating mga listeners, for some of you, we do hope na ma-interest kayo dito sa parasite work. It's very important work, food safety, a big concern, emerging concern. So yeah, I do hope you'll follow the steps of some of our, or not all of our speakers. Magagaling po silang lahat. And the future is bright, ika nga, when we talk about science and technologies in the Philippines, patungkol pati sa fishery science. So also, patenkyuhan na rin natin yung team ng training section and then their training division sa pangunguna ni Ma'am Norby, si Lay, of course, ang ating host, sila Joey, sila Dexter na nag-asikaso po ng CPD points, and to all, si Tito B at si Tonet, part po ng training section team. Also a great team sa NFRDI, hardworking and very dedicated people sa training section. So I guess yun lang po yung aking welcome remarks, ang aming welcome remarks. Ika nga watch and learn, enjoy the talks, ask questions, pick the brains of our very, very good speakers. Karamihan po sa kanila, if not all of them, ay medyo ka-age po natin. Ayun, ka-age natin. Ay ka-age nyo po sila, no? So I'm sure you'll be able to discuss things sa language na makaka-relate po ang bawat isa. Ayan. So again, this FishKwela training is for you. Andito lang po kami to continuously provide for the necessary information, new information, especially new technologies that will help all of you guys, that will help the sector, and of course help our country towards nation building. So good morning po sa lahat. Magandang umaga, and thank you for joining us for today. Salamat po. Bye-bye. Back to you, Lay.
Ayan, thank you very much po, Doc. Ayan, maraming salamat po for giving us time po. Alam namin napaka-busy nyo, pero napaka-accommodating talaga ni Doc Muji. Nox. Alright, sige po. We will resend po later on yung attendance link para po mahintay pa po natin yung ibang mga attendees. Isisend po namin dito po sa chat box later on. So please be stay tuned, no? Sa atin pong mga sa ating mga attendees na nandito ngayon. Ayun, i-resend po namin later on yung link. Okay, sige po. So with that, nandito na tayo sa exciting part ng ating mga lectures na. Ayan, sige. So for our first lecturer po, papakilala na po namin ang ating first lecturer. So our first lecturer is a researcher at the Kyoto University Graduate School of Global Environmental Studies. She obtained her Doctor of Philosophy in Environmental Sciences in 2023 at the University of Shiga Prefecture in Japan, and her Master in Environmental Sciences in 2020 at the same university. Among her recent research works include the redescription and molecular phylogeny of the nematode Megalobatrachonema nipponicum, addressing the taxonomic confusion of the trematode Mesocelium, discovery of 2 new catlanid species, the Nematoda Cosmocercoidea, parasitic in salamanders of the genus Andreas, first report of post-cyclic transmission in trematodes, Diogenes lacustris, first survey for helminths parasitic in hybrid and introduced giant salamanders, discovery of a hemorrhoid trematode, Bacteria maculae, uniformis, ready, and UV hatchery. Also served as in a 50 US symposium. She presented her work Evaluation for Alien Species, a Parasitology. Welcome, Dr. Karin Chuchida. Right, hang on po. We just have some minor difficulties sa technical aspect natin, but we will be playing Dr. Chuchida's presentation.
Okay, Lecture 1. Introduction to Trematodes and Nematodes. Essential Concepts and Knowledge. Presented by Karin Tsuchida. First, I introduce myself short. I am from Kyoto, Japan. I work for the parasitic elements of wildlife, especially amphibians, reptiles, and aquatic organisms. In my master's degree, I have been in Argentina and studied tremators of freshwater fishes in Patagonia. In my PhD degree, I studied nematodes of Japanese giant salamander and the tremators of Japanese urodilans. Now, I am a postdoc researcher at Kyoto University and a visiting researcher at the University of Shiga Prefecture. I have been in Philippines a few times to join the research project in Kasekuna. This time, I came to UST to join class as a visiting assistant professor. Okay, let's start the lecture about nematodes and tremators. First, I would like to ask you, what is a nematode? When you hear the nematode, you expect a thread-like animal, and someone becomes uncomfortable to watch their moving. It's correct. We call animals belong to the phylum Nematoda as nematodes. The name nematoda is derived from thread-like. They are triploblastic and symmetrical animals with a pseudocoel. Recent phylogenetic tree analysis shows that nematodes belong to ecdysozoa, and they undergo 4 modes before reaching adult. Nematodes do not have respiratory organs and a circulatory system. Marshall points out the cephalic end of nematodes and lead to the straight digestive system. The alimentary canal continues the anus in females and the cloaca in males. Ventral gland serves as the excretory system in nematodes. Their mouth and buccal apparatus vary depending on the diet. There are bacterivorous, scavenger, herbivorous, carnivorous, and nematophagous in nematodes. In general, nematodes are dioecious and have a sexual reproductive system. Occasionally, parthenogenesis. Some taxa of nematodes also include the hermaphrodite from hermaphroditic period. Mating and fertilization occur in the uterus of females. Their sex is not necessarily determined by genetics. How many species are in nematodes? Today, approximately 30,000 species have been described in the world. However, the total number of species is estimated to be 1 million or more. That means there are a lot of undescribed species in the world. About 75% of all nematodes may be free living. The rest of the 25% has a parasitic life cycle. So, most nematodes are free living in marine, freshwater, and terrestrial. We can find nematodes everywhere on the earth. One of the most famous nematodes is Caenorhabditis elegans for the C. elegans. This nematode is famous as a multicellular model organism because C. elegans has a similar structure to higher forms of life. C. elegans provides complete genome information. The present species also play an important role in testing cancer cells from urine samples. C. elegans is a free-living nematode and not a parasite. One of the most common thread-like animals is a horsehair worm. You can find them in the liver, and they come out heap of insects or become a mass liberally. They are parasites. Are they nematodes? The answer is no. The horsehair worm is one of the nematomorpha, and nematomorpha is a different phylum. Nematomorphs exactly resemble nematodes. How do they differ? All nematomorphs live in marine or freshwater, and most of them parasitize insects. I do not explain their body structure, but it also differs from nematodes. Okay, return to talk about nematodes. Nematoda is a huge group and has a marine origin. In each taxa, the acquisition of a parasitic life cycle has independently occurred. At least 15 instances of acquisition are estimated in the recent study. All parasitic nematodes may have a terrestrial or limnic origin. Here, I talk about parasitic nematodes. We can find parasitic nematodes in almost all vertebrates and invertebrates, also plants. If nematodes are ectoparasitism, they parasitize the body surface, gill, or skin of hosts. If nematodes are endoparasitism, they parasitize the organs, body cavity, blood, or inside of hosts. I show here is a true example of the life cycle of parasitic nematodes. Some nematodes need only one host in their life cycle, while others need several hosts to complete their life cycle. Next, the basic knowledge of identification. Morphological identification of nematodes is extremely difficult because useful characteristics of nematodes are usually so tiny. Before focusing on morphology, the first information is helpful to identify what it is. For example, host species, host collection site, infection site of nematodes, stage of nematodes, and sex of nematodes. We do not have a good visual book to look for nematode species, so we have to get differences to compare the descriptions with specimens. First information is significant to find optimum differences promptly. Let's observe morphological characteristics. The morphological characteristics of males are so useful to identify at the species level. chitinized spicles, well-known, and polar poplar are reproductive accessories. Those shapes, size, numbers, and arrangements are key morphological characteristics. Genital organs of females are also effective in identification. The position or ratio of the vulva, the form of the uterus, shape or size of the eggs are key morphological characteristics. Buccal apparatus is an important feature to identify. The morphological characteristics, regardless of sex, are also useful in hermaphrodite forms. Development, shape, or number of leaves, teeth, cephalic papilla, armpit, buccal cavity are key morphological characteristics. Recently, the combination study of both morphological and molecular identification are becoming a common approach in taxonomical study of nematodes. Next, I talk about trematodes. What is a trematode? Trematodes are the animals belong to the class Trematoda. The name of Trematoda is derived from aperture-like. They are triploblastic and symmetrical animals without body cavity. Trematodes generally have two suckers to attach hosts. All trematodes have an obligate parasitic life cycle. Trematodes usually need several hosts to complete their life cycles. Morphological characteristics extremely differ at each stage of development. The life cycle of trematodes includes both asexual and sexual reproductions. In general, larval trematodes asexually reproduce in the first intermediate host and release a lot of cell carrier. In the adult stage, most trematodes are hermaphrodites and sexually reproduce eggs. Lineage of trematodes secondary the complex life cycle of trematodes has evolved from a simple and direct life cycle. Ancestors of trematodes are estimated to parasitize vertebrate hosts. They added intermediate hosts in their life and made the life cycle complex. Trematodes generally utilize three types of hosts during their life cycle, or some of them have short life cycles. More than 20 instances of truncation independently occurred in more than 32 families of trematodes. Trematoda consists of two subclasses. Torreya, and Digenea. Aspidogastorea is a small group and includes about 80 species. Digenea is a large group and includes about 18,000 species. For the identification of tremators, the first information is also important. We can get a lot of fundamental information like host species, collection site, infection site of tremators before observing morphological characteristics. This information supports you to know what the species is. Briefly, I show the morphological characteristics of tremators. In adult specimens, most are hermaphrodites and have organs of both sexes. Terminal genitalia, ovarian complex, testes, uterine testes, vitelline muscles, and eggs are significant parts for identification. Other organs, for instance, pharynx, sucker, ceca, caica, have also useful features. The morphological observation for cecalia is important to reveal species and phylogenetic relationships. More than 10 morphotypes of cecalia are generally recognized. These morphotypes are classified by tail shape, the number of suckers, the presence or absence of eye spot, and others. The Frenzel formula is also a useful characteristic to identify. In recent years, the combination study of both morphological and molecular identification has become a common approach in the taxonomical study of tremators. Through this lecture, can you find your interest in nematodes and tremators? Not only in the medical area, but also in biological area. We don't know the fundamental information yet about species of parasitic nematodes and tremators. Let's dive into parasitology in wildlife to know what the parasite is. Thank you for listening.
All right, that was Dr. Chuchida for her presentation for this event. So, ayan po. Any question po from our participants regarding Dr. Chuchida's presentation? And we have here Doc Karin. Hi, Doc Karin. You still there? Ayan. So, Doc Karin will be answering questions po from our participants po regarding her presentation if there are queries po. So just to start the ball rolling, Doc Karin, what started your interest on parasitology?
In my case, my hobby, one of my hobbies is aquarium of freshwater fish, and I experienced a lot of fish disease. So first, I want to remove all parasites from fishes.
And have you also conducted parasitology studies relating to fisheries?
In Japan, focus on herbs, but in Argentina, I performed the systematic and experimental infection of trematodes of freshwater fishes.
All right. Currently po, where are you today? Are you in the Philippines?
Today in Philippines.
Yeah, at UST. You're still at UST.
Yes.
Yes. At UST po, what's your current studies? What are you currently studying on?
UST, I support their study for cell carrier and also the parasitic elements of other vertebrates, especially herbs, I think.
How are you liking Philippines so far?
Yes, sir, that's good, but sorry for my throat condition is really bad. So.
All right. Thank you, Doc Karin. Any question po from our participants? If ever, Doc Karin, can we... All right, may question from UP Visayas. Okay. Hello, Doc. Have you encountered endoparasites infecting the gonads of fish? Can you provide proper protocol on the collection and its identification or storage?
So the method for the fixation of the specimens is really different depending on the taxa. So if you get the nematodes, you first boil a little bit to become straight the specimens, make the specimens straight in the vial, and exchange from the exchange into the 70% of ethanol. But there are trematodes for molecular or for morphology. We change the style of fixation to stain or just tissue extraction. So really case by case, I think.
Thank you, Doc. We hope that that answered your question. Okay. Next, thank you for that nice presentation, Doc Karin. My question is, what are the usually affected species of nematodes and trematodes? Is it marine species or freshwater fish?
So affected species mean they're pathogenic to the animals, right? Maybe so. Uh, mm, mm,
Based on
so
your studies conducted so far, what species are the most affected? Are there more marine species or other more freshwater fish species affected? Doc.
affected means they're pathogenic to the host. So mm, most of the parasitic elements is not so. They don't have any symptom if they infect to the hosts because if they have no merit to die host. But sometimes to go to transfer the next host, they want to predate the predator to the host. So they manipulate or change the color of the fishes in marine or freshwater both. So depending on the infection site or depending on the transmission strategy, they change the symptom or absence of symptom. And I'm sorry because I'm not specialized to the ichthyological fish. especially symptomatic form. So I'm not sure, but also if they don't have any symptom usually, but they infect strongly, so they have a lot of population inside the host. They affect sometime, so for instance, the digestive tract symptom or something like this. Yes.
All right. Thank you, Doc. Next question would be, I would like to ask, do nematodes and trematodes release toxins or enzymes, or do they have significant effect on egg production suppression in aquatic organisms? This is from, I think, from our YouTube viewers, from our YouTube participants. Yan. I would like to ask, do nematodes and trematodes release toxins or enzymes? And do they have significant effect on the egg production suppression in aquatic organisms?
If the nematodes penetrate style for the transmission, so the infection of host fishes, if they penetrate the skin of them, the fish damaged in the skin or scale, the body surface. So such time there are slightly more risk to infect the bacteria or fungi. So second infection risk. But there are, I think, trophic from the trophic transmission, and so because there are a lot of parasites, but there are most of them are non-toxic. But there are sometime before I said they have a strategy to transfer promptly to the next host. So they change their movement and their coloring of the host to standing to the predator. So sometimes have a visual effect. So it's such a symptom is also not good for the human product, human activity to sell the fishes, I think.
All right. And another question from YouTube Live. How do you analyze parasites from water samples?
Water samples. So you mean to search the cercaria or in the free living stage of parasites?
Well, I think that the latter,
The one. It's really difficult because recently we can use the environmental DNA to search from the soil and the water directly, but it's really depending on the concentration of the DNA, and also there are a lot of undescribed species in nematodes and trematodes, and there are also other fungi and bacteria is there possibility to detect as their DNA sequences with other environmental DNA. So that's such species, such other organisms also disturb our detection of specific nematodes or trematodes from water samples. And also Cercaria form is very distinct to the species, but their nematodes, so free living nematodes, so before adult stage, it's very difficult to identify morphological because they are still molting, and they get gradually their genital organs and their other morphological characteristics to identify species. So third stage or second stage, first stage, yeah, every stage of larval nematodes is really difficult to identify. Yes. So, but if we have the DNA sequences, we have a chance to detect the environmental DNA, but there are a lot of possibilities to detect other DNA from nematodes, termites, and also bacteria or fungi.
So, and another question from the Q&A chat box: is it difficult to identify or distinguish sexual and asexual stages of trematodes? Did you find any difficulty assessing or distinguishing this species that you have already studied?
So one of the difficulty to identify the different stages of trematode specimens is morphological characteristics is really different from each stage. So first, we cannot directly consider or regard as the same species from the larval asexual reproductive stage to the adult stage. Yes, but we have the molecular identification, easy pattern for the systematic identification. So we can now just from the tissue we can to connect the life cycle for one species. So for the described species, it's easier, but there are undescribed species and only each stage, if you find, it's difficult to direct connection you can find.
All right. Maybe last question for this presentation, Doc, and I would just like to ask, after this presentation, can we still like send you queries or any questions from our participants? Po? Where can they contact you?
Yes, of course.
Where can they contact you? Can they find you any social media page or any email address that we can like message you?
Yes, yes.
So
Yeah,
we can even like
maybe,
just message you
uh,
via email.
yes, my email is better for me because I'm so lazy user of SNS. So,
All right, Doc.
yes.
All right. Thank you very much, Doc Karin. Thank you for
Thank you so
gracing us in
much.
this FishKwela activity.
Okay.
Ayan.
Thank you so much, Paul.
Thank you po. And with that, we will now be okay, keep the questions coming. I-forward po namin yan kay Doc Karin for further discussion sa inyo personally, no. I-send po namin sa kanya yung mga questions ninyo via email maybe. We can like connect you to her para masagot niya yung mga questions na ni-raise ninyo. Okay. Thank you, Doc Karin, and thank you po sa atin pong mga participants. We will now go to the next topic of this FishKwela activity. So, the next lecturer, okay, the next lecturer is Doc Janelle Ragacad. So, Doc Janelle is a Balik Scientist awardee. She has finished her PhD in Environmental Science at the University of Shiga Prefecture in Japan, and her Master of Science in Microbiology at the University of Santo Tomas in Manila. So, wow, she graduated cum laude during her master's studies. So, currently she's a Balik Scientist and is under the National Fisheries Research and Development Institute Her study, uh, the study that she has contributed to is the Picard-funded project Ecology and Cercarial Production in Tropical Freshwater Ecosystems, host parasite connections of trematodes in the Cagayan River. So, research assistant po siya sa project na yun. So, her skills include DNA sequencing, real-time polymerase chain reaction, gene annotation, developing primers and probes, parasitology, phylogenetics, microbial analysis, molecular biology, mitochondrial genomes, and she's also fluent in our programming language. So, yan. Due to her work, she has received various scholarships and recognition from the Japanese government scholarship. Then the Department of Science and Technology, DOST, SEI, ASTRODIP, NSC, yan. Then she has also published her researches. Some of them are the trematode species detection and quantification by environmental DNA qPCR assay in Lake Chany, Russia, characterization of the complete mitochondrial genome of Plagiarchis multiglandularis, comparison with the members of Siphidiatan species, and phylogenetic implications. The complete mitogenome of Echinoparifium aconiatum, and a comparison with other Digenian species. Then Aeromonas veronii and Pleiosomonas shigaloides isolated from Glossogobius species in Lake Sampaloc, Laguna. So she has also presented her researches in various conferences and has attended also various trainings so she can develop her skills. So ladies and gentlemen, we would like to welcome now Dr. Janelle Lara J. Gakat.
So, good morning, everyone. Welcome to our FishKwela webinar. Today, we're so excited to have all of you here as we're going to delve deeper into the fascinating world of parasitology. Whether you're students or you're researchers or you're just simply passionate in studying about this topic, this FishKwela webinar is for you as we will have a comprehensive look in parasitology from the basic concepts to the more advanced techniques. So, as much as possible, we want you to have many takeaways for this webinar. Okay, so for the lecture 2 is about exploring parasitology, an introduction to the morphological and molecular techniques. But before we delve into our agenda today, I want to introduce to you our research laboratory. Currently, I'm part of two projects. The first project is the bilateral exchange research program between Russia and Japan, and the other one is DOS joint program between DOST and JSPS. Both of these projects they focus on the environmental DNA analysis for trematode monitoring. Okay. So, mamaya mas i-discuss ko yung projects namin sa molecular techniques. I-introduce namin kung ano yung ginagawa namin sa recent project. Okay, the first question is, why are we all here today? Why do we have this FishKwela webinar? Specifically, why is it important to study parasitology? We will have some examples. Okay, first example is, did you know that to spark your curiosity in studying this topic, did you know that the recent Nobel Prize was given to Dr. Victor Ambros and Dr. Gary Ruvkun? So they were just studying about this small worm, or specifically C. elegans. It's a nematode. So from this small worm, it led to a big breakthrough. So basically, ang inaaral nila yung development ng isang nematode. Yung dalawang mutant strains. Isang strain ng nematode lacks Lin4 gene, the other nematode lacks Lin14 gene. These genes, they are responsible for the timing sa pagdevelop ng isang nematode. But it is expected if you lack these genes, merong magkakaroon ng developmental defects. So as you can see here, yung normal, ayan. So, okay yung movement niya, yung pagdevelop niya. But here, when you lack Lin4 or Lin14 genes, expected that they will have developmental defects. So, what they're studying is ano ba yung connections between these genes. Okay. So, what they found out, to cut the story short, is what they found out that yung Lin4 gene ay may complementary sequence sa Lin14 genes. So, this cemented the existence of a non-coding RNAs called microRNAs. So before, hindi yan pinapansin sa studies. Na this microRNA, that this can treat genetic disorders such as cancer, autoimmune disease, neurodegenerative disease, and can also be biomarkers for diagnosis of illnesses. So from that small worm, na cement yung existence of isang microRNA na dati neglected lang siya. Ayan. So, na-spark na ba ang inyong curiosity? Another example is, I want you to picture a scenario. This is really happening in real life in Africa. There's an infant dying because of an infection caused by mosquito-transmitted parasite, which is the Plasmodium falciparum. So why is this happening? So either their home is too far from the clinic, or they just simply lack the resources to buy the medicine. That's a common scene in Africa na average on about every 2 minutes may namamatay. Which is, na-realize natin na isa pa ring challenge, part ng challenges itong mga disease na ito, such as malaria. It's still a formidable adversary. Not only that, 90% of the children there in Africa are infected with schistosomiasis, disease caused by parasites, specifically the schistosome species. It's transmitted in Africa, but these parasites, they're not only found in Africa, but they're dispersed in different parts of the world. Ayan. Actually, estimated niya na 1/3 of Earth's species are parasites, with approximately 75 to 300,000 ang nag-i-infect sa vertebrate hosts. And as you can see here, ito ay isang table that these parasites infects millions of people and causing thousands of deaths per year. Like what we've discussed, yung malaria, na infecting 214 million, and ang pinakamataas na deaths per year, 438,000 deaths per year. So, ganon yung impact or yung negative impact nitong parasite sa human health. Another example is, sino po ang who here loves to eat sushi? So, yan. So, I myself loves to eat sushi, raw fish, raw salmon, yan. And it's not a secret that naging popular yung Japanese cuisine nowadays. Yung sashimi, sushi, yon. But, ito palang mga raw fish na to, mga sushi, sashimi, they're suspected to cause fish-borne parasitic zoonosis. So, yan, para aware lang po tayo. Paano tayo nai-infect paano tayo nagkakaroon ng sakit through eating the sushi or sashimi? Because ang example niyan is yung Diphyllobotriasis. Ang intermediate host niyan ay salmon. By eating the raw salmon, we can acquire this D-latum parasite. Another example is a case from a patient. Nakita sa x-ray niya na nag-travel yung parasite to its lungs. Yung Paragonimus westermani. It's a parasite causing a paragonimiasis. So, 2 months before, kumain siya ng sashimi. And after 2 months, nakakaranas na siya ng symptoms. Ang intermediate host nito ay crabs or yung dahil kumain siya ng sashimi. So, this fish-borne parasitic zoonosis, naging endemic siya in Asian countries. Not only the Philippines, not only Japan, but in many Asian countries. Because of this, we come to realize na these parasites still exert a powerful grip, not only to humans, but also to animals and plant species. But they are not only causing negative impacts on human health. They could be beneficial also, such as they play key roles in food web and energy flow within ecosystem, such as they're important element of the ecosystem and population dynamics, and they can be indicators of environmental stress and biodiversity. Now we know that they have this ecological significance, zoonotic significance, and they have negative impacts on human health. And But how can we study parasitology? It can be studied at many different scales, such as molecular interactions, yung biodiversity, kung paano makadiagnose ng disease, yung implications for human health, paano makadevelop yung new vaccines, drugs, or control strategies. Ayan. So the greater understanding, the more we study these parasites or yung responsible organisms, the more we can develop new vaccines, develop new drugs, to have control strategies. Yan yung importance of studying this. Unfortunately, kahit sa lahat ng diniscuss po natin today, meron siyang high impact sa ecological importance left out in the investigation. And still lacking ang systematics, it's still incomplete because there are challenges on the identification of trematodes, specifically when using only morphological characteristics as Cercaria. Thus, the rise of morphological and molecular techniques. Kaya, it's important to study it. First is the morphological. Dun muna tayo sa aspect na yon, which is part of our topic today. And we have multiple tools nowadays, but we'll dive first into the morphological identification. How to isolate trematodes. So it's important that we know first the life cycle of these trematodes. Paano ba natin na-acquire yung parasites tayong human? So, next-start po yan sa embryonated eggs that are passed in feces. So, nasa feces siya, and then may ingest ni snail. The snail will ingest the eggs, and then mag-hatch si egg inside the snail, and then mag-release si miracidium. Si miracidium will develop into generation of sporocyst, develop into cercaria, magkakaroon ng head, and then ng tail, and then may cercaria, mag-release si cercaria sa snail, and then may ingest or pupunta siya sa flesh ng fish, and then tayo, kinakain natin yung flesh ng fish, makukuha ni human yung parasite. Yan. So, ganun po natin na-acquire or yung life cycle ng trematodes. So here's one of my studies. We collected fish samples, so aquaculture, fish samples from Lake Sampaloc. So, nag-collect kami ng fishes, and then we dissect the fish, and then in-isolate namin yung gut, and then inopen namin yung gut, and then yung parasites. So here, so you can see, kapag na-isolate mo na yung parasite, dun sa intestine ng fish, i-open mo siya. So, usually, mahirap siyang kunin because yung attachment ng parasite ay because of the oral sucker and the ventral sucker. So, dahil maraming nutrients silang nakukuha sa intestine, andun basically maraming parasites dahil dun sila nakakakuha ng nutrients. If you already isolated the parasite, meron kaming hina-half namin yan. Half for the morphological, half for the molecular identification. So, for the morphological identification, nagpe-prepare kami ng slide. We have these different reagents. We have the hematoxylin, different series of alcohol for the dehydration. We have the xylene. We have the Canada balsam. Lahat yan essential for staining and mounting the slide para ma-preserve at magkaroon tayo ng permanent slide. Now, if you're finished with the permanent slide, ganyan yung itsura niya. Mas madali mo siyang ma-identify, makikita mo yung morphological characteristics niya. And then some, we can preserve them in ethanol para sa molecular identification. Ayan. Ano ba yung mga different factors bakit high prevalent yung parasites and host? So here, our fish sample is Glossogobius aureus from family Gobidae. So because of its omnivorous feeding behavior and benthic life cycle na lagi siyang nasa baba ng lake, it is more highly susceptible to infection by parasites. So it can ingest more intermediate hosts, more eggs. Kaya when you open or dinisect mo yung goby fish, ang dami niyang parasites. Next is, as we discussed, and di ba, yung first intermediate host can be snails. We can also isolate Cercaria. This is one of our projects from Russia. So we collected snail samples from Russia, and then we observe the shells and para ma-identify mo din yung host niya. And then there are two ways in order to isolate parasites from the snails. It's either you crush them here, as you can see here, or let the Cercaria shed by putting it in the aquarium. But here, ang pinaportray or pinakita po namin ay yung pag-crush ng snails. When you crush the snails, the Cercaria will be released, and then you can now prepare the slides for it. Okay. So, ito po yung ilalagay mo siya sa aquarium. So, meron siyang different conditions. Kailangan yung requirement for the light, for the temperature para mag-release yung Cercaria. And this is the Cercaria. We can now identify it using the morphological characteristics. Okay. So, ito po yung project namin with Dr. Muji and Dr. Briones, and kasama rin po namin si Dr. Karine and Dr. Marcaida sa Caseknan River. So, what we found out are yung Cercaria, we isolated Cercaria from the snail samples, yung Haplorchis species. Has potential for zoonosis na nakakapag-cause ng intestinal disease. And we found out na itong Caseknan River ay tourist spot din pala to. So that's why it is that's why nag-focus kami in studying this collection site in Casecnan River. And after you mounted the slide or fixed, you stay in the slide and magkaroon ka ng permanent slide, you can now see, ito yung isa sa mga slide ko na minount ko. So you can see the different parts of this trematode. So from its mouth, oral sucker. So very important yan for the morphological identification, especially when you use the taxonomic keys. So yan yung parts ng trematode. You can see here the ito yung for attachment, yung oral sucker and ventral sucker. And here you can see the vitelline follicles. That's all important para ma-identify mo siya even to the species, the genus level. Here is an example of the Haplorchis species na na-isolate natin sa Casecnan River. Yan po yung sa mga study ni Ms. San Diego. Now, let's learn how to use the dichotomous key. So parang after mo ma-mount yung slide, after mo ma-observe siya sa microscope, what's next? So you will need to learn how to use the dichotomous key. So let's have exercise para marami po tayong takeaway for today. Okay, so here's a simple example. So dito muna tayo sa madadaling parts bago tayo mag-proceed dun sa Cercaria. So here's how to use the dichotomous key. So first, you need to start sa 1A, always sa 1A. So does the monster have legs? So meron po ba siyang legs? Wala. So pag wala, you proceed to 1B. The monster does not have legs. So that's correct, right? So if that's correct, go to 5. So monster has one eye. That's incorrect. And the monster has more than one eye. So tama because dalawa yung eyes niya. So go to 6. So monster has 6 or more eyes. Nope. So go to 6B, monster has 2 eyes. So this is a winger species. Another example. So baka kaya nyo po sagutan yung another example natin. You can type your answers below. Ayan. So another example is ano po to? Anong species ito? So does the monster have legs? Meron. So go to 2. The monster have 5 eyes. Incorrect. Go to 2B. The monster does not have 5 eyes. So correct. Go to 3. Monster has horns. Correct. So it's bullhorn species. So that's how you read the dichotomous key. So let's proceed naman na alam na natin yung basic technique how to read the dichotomous key. So let's proceed dun sa medyo challenging part, yung Cercaria. So in identifying Cercaria, we need to use the keys by Shell 1985. So here is our isolated parasite. Makikita nyo yung, you can see the mouth, you can see the oral sucker, the ventral sucker, the excretory vesicle, and yung wala siyang tail. So, the tail is absent. So, correct. You need to go to 2A. Cercaria develop into develop in branched porocyst. So, thin yung vesicle niya. Thin ba yung vesicle niya? So, no. Go to 2B. Cercaria developed in Rege in aquatic snails. Its excretory vesicle has thick cellular wall, so it's a mutable cercaria. Yan. So that's how you read the dichotomous key. But this morphological identification, there are a lot of challenges in using it per se. Some may similar morphology. It's also time consuming, meron tayong limited reference material, and sometimes poor yung specimen quality sa pag-preserve. We have cryptic species and yung developmental stages niya, meron siyang cercaria, may adult trematodes. So there are a lot of challenges that we can face in using this method alone. Thus, the importance of molecular identification. Ayan. So, ayan, that's the rise of molecular identification. So, this is the part 2 of our lecture 2, yung molecular identification. Okay. Because of the challenges, some groups of parasites, they can be tractable, such as the tapeworms, the trematodes, the nematodes, and acanthocephalans infecting vertebrates. But some species, especially those that are infecting plant species, hindi sila, it's difficult to track them. So one of our major shortcoming actually is our inability to our shortcoming here on Earth is our inability to say with confidence how are how many species are there really that exist on our planet. Some researchers say it's million to trillions, but we can never be sure kung gaano man kadami. We can never be that confident. Why? Because there are lack of thorough sampling of many habitats, there are many presence of cryptic species, and potential loss of species na mas mabilis nag-extinct than we even discover them. They are small, they can be easily overlooked. Um, it's not easy to isolate them or it's not easy to culture them. So that's why um, dito pumapasok yung importance ng advanced molecular techniques such as eDNA and metagenomics. It's a characterization of genetic material recovered directly from particular environments such as the soil, water, and even the air, without the need to culture them. So, I tried metagenomics when I was studying in Japan, which is sampled water and then finilter yung water, and then after that extracted the DNA and sequenced them. Then if you know some the bioinformatics or the coding, malalaman mo na lahat ng presence ng bacteria, lahat ng presence ng parasites from that simple sample of soil, simple sample of water, and simple sample of air. But here we in this project or in this discussion, we focus more about eDNA. Itong eDNA, it's a very sensitive and powerful diagnostic tool for detection of trematodes. It target to find DNA in environmental samples. So, yung eDNA, it's a genetic material shed by the organisms from the skin, nagsished ng tissues, the mucus, the urine, the feces that we can collect from the soil, from the fresh water, and even from the ocean. It is very useful for parasite kasi sobrang hirap ma-locate and accurately identify. It can also be quick and cost-effective detection for trematode species. So this is what we use here is quantitative PCR or the qPCR. Ito, as we know, during COVID, lagi nating narinig PCR, qPCR, yan. So it's a gold standard method to test patients for viral infections such as COVID-19 to detect infections with pathogens. We can also use this for parasites naman. So we all know kung ano yung process of PCR, overview of polymerase chain reaction. So in a thermal cycler, what's happening there when we have this master mix na para lang ma-recall natin so we can recall what's happening inside a thermal cycler, what is happening inside qPCR. If we have this master mix, we have the DNA sample, we have the primers, we have the nucleotides, the Taq polymerase, siya yung for extension, mix buffer, and the PCR tube, dun mo ilalagay. And then, what the thermal cycler is doing is it will increase the temperature to 95 degrees. Di ba ang DNA natin is double-stranded siya. So, pag tinaas ni thermal cycler yung temperature, magse-separate siya. That's what we call the process of denaturing. So, pag naghiwalay yung DNA, double-stranded DNA siya naghiwalay, bababa na naman ni thermal cycler yung temperature, and then, darating si Taq polymerase, and then, ay mag-aanil muna, and then, si primers, yan. So, the forward and reverse primers, and then, darating si Taq polymerase, and i-extend niya hanggang mag-produce ng millions of copies of DNA. So, yun yung PCR. Produces millions of copies of specific sequence. So, in the qPCR, we have this, what we use is cybergreen or yung Taqman assay probe natin. In my study, I use Taqman assay kasi there are pros and cons in using cybergreen and Taqman assay. So, sa cybergreen, it's cheaper and yun yung pros niya, pero hindi siya specific. Ayan. So, mag-extend siya. So, sa Taqman naman, it's specific but it's more expensive. So, if you design your primers and probes, magbabay lang siya din specific na species na designan mo ng probe. So, ayan. So, yung fluorescence, yun yung magsisignal. Kaya pag sa qPCR na naka-amplify siya, we will discuss that in detail sa later ng ating lecture. So, one of our speakers will also detail the mitochondrial genome. So, si Professor De La Luna. So, it's also important gusto ko lang din i-mention that it's important for taxonomic status, species diversity, and identification. For the methodology of our eDNA study, so first we collected water samples, snail samples, and then we designed primers and probe, determine its specificity, the specificity of the primers, and we use qPCR, evaluates its efficiency using the water samples. Ayan, so ito yung detailed part. We collected the water samples and then filtered the water samples in every 500 ml. Finilter namin siya using the glass fiber filter. So, dyan nagko-collect yung eDNA. After which, na-mention natin kanina kung paano makapag-isolate ng Cercaria. We extracted the DNA of those parasites. And then, we sent them for sequencing. And then, after which, we will receive the assembled mitochondrial genome para makapag-design tayo ng primers and probe that is needed for qPCR. So, we need to annotate them. In annotating, malalaman natin yung mga protein coding genes. So, in designing primers and probe, para meron lang kayong gist or meron lang kayong takeaway, sa molecular techniques, here is how to design primers and probe. After annotating the mitochondrial genome, we can use some softwares, Primer Quest, Primer Blast, in designing primers and probe. Usually, sa Cox 3 or sa Cox 1 to 3, ako nakakakuha ng best primer and probe set. Ayan. So, when you already design your primer and probe, and then you will now use qPCR. Yan, ito yung qPCR. And then, kapag positive siya, mayroon siyang amplification curve. Pag negative siya, flat. Flat lang siya. And here, in order to know the efficiency, you need to have your standard curve also. Ayan. So, yung kailangan din i-optimize natin yung conditions, how many cycles, yung temperature niya, annealing, melting temperature niya. So this is our optimized condition. We also check the specificity using the electrophoresis. How do you know if it's specific? So here is an example of a specific design primers and probe. Ayan. So yung M-ANSIC, specific lang siya for M-ANSIPs, specific lang siya for RNA. Hindi na-amplify yung non-target species, yung ibang species, negative siya. But here you can see, with our design primer and probe for plate Jorkis, meron siyang amplification but sa P. elegans. Pero it's safe to assume that it's genus specific because of very high CQ value. So usually ang greater than 30 of high CQ value, hindi na siya kine-consider na positive. But it is still safe to say na genus-specific itong primers and probe. In this agarose gel, you can see that it's specific, visible siya, and wala siya mga primary dimers, walang PCR inhibitors. So, iisang band lang siya. How will you know if high efficiency, you have high efficiency of primers and probe? So greater than 0.980 yung linear standard curve, 90 to 105%, and consistent yung replicates mo. Here is the results with our eDNA samples. You can see from that water samples, positive siya. So, meron siyang na-detect na parasite. So, ganun lang siya kadali. Kapag nakapag-design ka na ng method mo, ng assay mo, magko-collect ka lang ng water samples, and then, ilalagay mo lang siya sa qPCR. And then, i-detect niya na kung positive or negative kung may parasites ba. You can use this in water samples, you can use this in soil samples, and even in fecal samples. We collected water samples from 13 sites. So, positive sa 9 out of 13 sites ang M-ANSIPs, 2 out of 13 sites yung RNA, and 7 out of 13 sites yung P. multiglandularis. So, from that simple water samples, you can already detect these 3 species. Ayan. Ano ba ang example ng failed experiment? So, yan. Ang daming primer dimers, ang daming PCR inhibitors, poor primer design, and maybe your PCR mix is not appropriate. So, ang daming bands. Hindi siya specific, and then wala siyang amplification curve. So, you need to troubleshoot. We also established a calibration curve. So, kung positively linear correlated ba yung microscopy counts versus the qPCR count. So here you can see the linear regression showed a positive linear correlation between the microscopy count and the qPCR count. So in every 500 na kinolect naming water samples, merong 11.80 cercaria or per liter, there's 22.48 cercaria. So ano overall yung contributions of this assay? So, and in this study, in this molecular method that we're learning now. So, in ecological aspect, it's useful and beneficial for ecosystem monitoring, measurement management. In taxonomy aspect, it's based for new markers. It will enrich the mitogenome database. And for medical, it helps diagnosis illness through the detection of eggs in the stools. Collect mo lang si fecal, karon ka, si stool, and then qPCR. Diretso na yon kung positive or negative. So, thank you for participating. I hope you learned something. Just feel free to ask if you need any clarifications. Thank you and God bless.
All right, thank you very much po, Doc Janelle, for your presentation. And yan, may mga questions na po tayo sa atin pong Q&A box. Doc Janelle, are you still there po? Okay.
Yes po. Hello
Hi
po.
po, good morning po, Doc Janelle. So, Doc, pwede po bang kwentuhan niyo lang kami konti doon sa naging study niyo sa Cagayan River?
Okay. So, yung Cagayan River po, yun yung ongoing project namin. Actually, lahat po ng speakers natin today is including Doc Muji, he's one of our collaborators. Kami tayong NFRDI, and then we are collaborated with JSPS, yung with Urabe Sensei, and Dr. Karin, Dr. Arvin, and Dr. Briones. So, yun po yung ongoing project namin na ito po ay originally pinropose po ni Dr. Briones, and now we're still continuing it. On our end, I am focused more on the molecular aspects. Sila Dr. Marcaida and Dr. Karin po, they are focused more on the morphological identification. And here, ang nireport namin is meron pong nakitang parasite trematodes um, sa snails and even sa frogs. But nag-collect sila ng fish samples. Unfortunately, walang nakitang parasite sa lahat ng fish samples na
Okay.
kinolect nila. Meron nakita na Haplorchis species. This is parasite that cause intestinal disease. So yun po, kinda alarming po doon.
And yung mga na-collect niyong fish species po, anong species po yun?
Hindi lang po, more on yung sa end namin ay snails. Sila Miss San Diego po or yung other part of the group, sila yung assign po dun sa fish species dun sa Caseknan River. So i-double check ko lang kung ano yung specific na fish species na na-collect po nila.
Ayun, opo. Kasi medyo interesting study din po siya for tawag dito, yung mga gusto mag-study din doon sa area na yun and sa species no, especially sa fisheries. So, questions po from the Q&A box. Yung first question po actually is based doon sa presentation nyo kanina wherein you tackle yung din sa Glossogobius species. So, in your sample species, how did you know that the fish are infected with trematodes, or what are your signs that it was indeed infected?
Okay. So, thank you for your question po. So, usually, kapag merong presence of parasites yung fish, meron siyang visible dark spots, may mga cysts, even sa skin, sa gills, or dun sa fish. Meron we call black spot disease, meron changes of the behavior, the way it swim, disoriented yung swimming niya. But on my end, for the goby fish or the Glossogobius aureus, where we collected more than 100 samples kasi per month po yung sampling namin para makita namin yung number of parasites per month. Depende sa season, kung rainy season ba or summer, yun. Wala sa more than 100 fish species that we collected, I did not observe signs physically if there's parasites, wala siyang spots or Yun, wala siyang cyst. But when we open the when we open the fish and open its gut, so highly prevalent yung parasites. And specifically Opigoster minima yung parasites inside the goby fish. Ayun po. And winade din namin siya yung length niya, yung weight niya. So, nakita namin kung parang sa study na yon in-identify namin yung maraming parasites kung ano ba siya, yung smaller ba siya or bigger, yon. So, nakita namin na mas heavily infected yung mga maliliit na fish. It means siguro yung mga younger fish, mas ano pa, compromised pa yung immune system nila than the larger fish. Yun po.
Interesting, no? Kasi, especially yung mga ano, kasi baka hindi din natin alam, infected din yung sa mga aquaculture industry. So, ayun. Next question po will be, hi po, Doc. What reference po ang magandang gamitin for parasite identification?
Okay, so we have different keys as I mentioned earlier, yung dichotomous key na I hope you learned something from that exercise. And sa Cercaria, we use the keys of Shell 1985. Yun yung ginagamit natin for Cercaria. For adult trematodes, we use yung keys to the trematode of keys of Velasquez. So, yeah, and if you need these reference or keys, you can email us and we can, send you a copy para meron kayong guided reference for the identification of parasites. Ayun po.
Right. Next po will be, good morning po, Doc. Just to clarify, why is Cercaria isolation important? Is it because it is easily collected or easy to identify?
Why is Cercaria isolation important? Okay, so do you know in the Philippines that ito more on site environmental monitoring type, but in the Philippines, do you know that it's a major public health concern yung schistosomiasis. So I think you are somehow familiar to that many Filipinos are infected with this schistosome species and nakukuha yun sa snail samples. And yung snail samples, they are intermediate host. Ang nakikita sa snail samples ay Cercaria. So, it's important to disrupt the infection as early as in the snail samples para hindi na ma-acquire ni final host or ni humans yung Cercaria. So, that's why it is important to isolate that para maging aware tayo heavily infected by yung snail samples dito sa area na to, may presence of schistosome species ba or that can cause disease. Like in our project in Casecnan River, we found the Haplorchis species causing intestinal disease. Is it easily collected? As I mentioned earlier, we have two methods. You can either crush the snails or prepare your aquarium so to wait to the shed of the Cercaria. So, ang mas madali doon ay yung i-crush mo yung snails, but it doesn't 100% confident na lahat ng Cercaria ay nandun sa pag-crush ng snails. Na some ay hindi pa ready yung Cercaria or hindi pa okay yung life cycle niya. Na-crush mo na yung snails. So, mas maganda siguro yung sa aquarium. And easy to identify as I mentioned earlier, yung keys to yung dichotomous key, yung kay Shell 1985. Yeah, if you read that key, yung exercise natin, I think yes, it could be easier for you to identify.
Right. Thank you for that po. Other environmental factors influencing the isolation of trematodes in aquatic ecosystems.
So, are there environmental factors influence them? Okay, so yes po, there are a lot of factors that influence the isolation of trematodes in aquatics ecosystem. So, kaya din 6 months din yung sampling namin para makita din namin yung different factors na nakakapag-affect dun sa pag-isolate ng parasites. So, For example, ang kinuhanan namin ay yung aquaculture. Aquaculture versus the wildlife. Ano ba yung difference kung ano ba yung mas highly prevalent? In aquaculture, it's more on controlled, it's more on dense, and then I think stressful yung environment for the fishes, for the host. So, mas highly prevalent yung parasites kasi dahil controlled siya, stressful yung environment, na immunocompromise yung host, mas nagiging mataas yung chance na maging susceptible siya sa parasites and mas mataas yung infection na magkahawaan sila ng parasites dun with that controlled environment. On the other hand, when it's wildlife for that environmental factors, um, hindi siya highly prevalent compared to the aquaculture, but mas mataas yung diversity of different species ng parasites dahil different biodiversity yung mga snail host, yung mga host na life cycle ay umiikot. Yan po.
Right. Related pa rin po sa environmental factors, Doc, you mentioned po na temperature is an important factor in the presence of Cercaria. Usually, what is the temperature range in order for Cercaria to be present or high in amounts?
Okay, so I think I mentioned that in my previous publication. Also, I learned that from our mentor, Urabi Sensei, na meron bang factor ang temperature in the presence of Cercaria. So, surprisingly, hindi na sa Cercaria, di ba, kailangan merong aquarium. When you set the aquarium, there's specific temperature, there's specific light na kailangan para mag-shed si Cercaria. And what 20 room temperature is appropriate para mag-shed si Cercaria. But there 27 to 32 degrees Celsius. So yun yung basically standard pa now, but I think it's still ongoing study dun sa range ng temperature. Ayun po.
Right. Siguro po last question muna na for this topic, Doc. Doc, medyo marami ito. Sabi niyo po, what will be most likely to produce good results with TAC or without? And did you include reference genes? Then, pa-include na lang po siguro on your answer. How do they select appropriate primers for qPCR? Some common causes of high variability in qPCR results, and how can they be minimized?
Okay, so it's about, I mentioned the pros and cons. It's more about the budget because if you have the budget, I really prefer the TAC assay than the cyber green. Pero kasi if mas cheaper kasi yung cyber green than TAC. And dahil ito ay project, we were able to use the TACMan probe. So the TACMan probe, it's specific. If you are just identifying your specific species, for example, for your schistosome species, kapag ginamit mo yung TACMan probe, ang ma-identify at ma-amplify lang niya ay yung schistosome species. On the other hand, if you use the cybergreen, marami kang pwedeng ma-identify doon, like mayroong possibility of the primer dimers. It's not highly specific. It's not highly sensitive. But with TACMan probe, I witnessed that it's highly specific, highly efficient, and highly sensitive. And what do you mean by reference genes? What we used is yung mitochondrial genome sequence na in-extract namin from our samples. And then when we were able to sequence that, I annotated the mitochondrial genome and dun ko binase, dun sa 12 protein coding genes na yon, dun ko hinanap kung ano ba yung best gene. And the best gene is usually from COX-1, COX-2, and COX-3. Dun mo makikita yung best primers. And how do we select appropriate primers for qPCR? It's more on a trial and error. There are a lot of softwares that I use, but I prefer coupled with softwares, I use manually check it then to design the primers and probe. Merong maraming reference sa mga studies. If meron naman na published na primers, you can use that already. So, what we did here is wala pa kasing primers itong mga species na design namin. Kaya, we designed, first designed these primers and probe. Some causes of high variability in qPCR results and how can they minimize. So sa qPCR results, as I mentioned, it's more on a troubleshoot, trial and error. So you need to optimize your qPCR conditions. You need to test its efficiency, yung water sample na kinolect namin sa Lake Shani. So kung positive ba yung amplification niya, we use the melting curve analysis if it's highly specific. And yung sensitive, highly sensitive, you need to use the standard curve. Ayan po. So we can discuss more that in detail. You can ask us directly po.
Right, thank you, Doc. May mga questions lang din po from YouTube participants po, ano. So, we'll discuss, okay lang po ba i-discuss pa po natin ng konti, no? Okay po. So, the first question from YouTube participant is there a chance that you mistakenly identified the parasite, or did you find it difficult to identify the parasite? If so, what did you do?
Kung nagkaroon po ng instance na mistake yung pag-identify ng parasites, and if na-mistake, what will I do? Yes po. So, on my experience, I'm not sure if na-experience ko na yun, but I'm thankful to my mentors, Dr. Briones, Dr. Misako Orabe. So, kino-confirm ko sa kanila after I identify it, and then I confirm it to them. So, through morphological identification. So, I think that's important na in order you to limit or to prevent those misidentification, or yung for misidentification, you need to confirm it with your mentors. And coupled with morphological identification is the molecular identification. That's why it's important na magka-partner po sila para 100% confident ka that this is the right species, the identified species. Yun. Kasi when pareho sila na, for example, Plagiorchis sa morphological, you use the keys to identification that it's Plagiorchis, and then when you sequence it through molecular identification, so na-confirm mo, so you can be 100% sure and you can publish your data. Yun. Yun yung laging hinahanap in a publication, na i-confirm mo na now through molecular identification. Ayan. And deposit in GenBank. Si GenBank din ang nag-a-approve if tama po yung species niya.
Sige po. And siguro panapos na lang po na question for this topic. So this is not related to identification, but how can consumers ensure that they do not get infected by parasites? Is cooking them well enough? Or are there species or stages which are heat tolerant? Thank you po.
Okay, so in bacteria, kasi 'di ba meron tayong mga ganyan kung thermophilic ba yung itong bacteria na to, namamatay ba? So what I discussed earlier, that mostly cases that are infected with schistosome species, Plagiorchis species, Paragonimus species, they usually eat the raw fish, yung snails, ayun po. So I think what would I suggest is to properly cook the fish samples, or yung snails, and yun. I think that's the importance of this study, na maging aware din yung mga tao when we identify parasite in this site, yung Kaseknan River. We need to inform yung people that are living there that this is present, so they can have this information, na maging sila alert or aware sila not to eat this raw fish or yung mga snails around the vicinity. Ayun po.
Ayan. Narinig po natin si Doc Janelle. Maraming salamat po, Doc Janelle, for your time. Para dito sa atin pong ParaMapping event. Ayan. Maraming salamat, Doc Janelle. And if ever po na may mga questions sila, i-forward po namin sa inyo, and you can answer them through email siguro, Doc.
Thank you.
Ayan. Sige. Maraming salamat po, Doc Janelle. Ayan. So, yan, ready na ang ating next speaker actually. Sige po, ayan. Pakilala ko lang po ng saglit ang ating resource speaker po before ko sa kanya i-turn over ang floor. So, our next resource speaker po is Assistant Professor Arvin Jet B. Marcaida, who is an accomplished researcher and educator with expertise in parasitology and environmental science. He earned his PhD in environmental science, majoring in parasitology from the University of Shiga Prefecture in Japan, where he conducted pioneering research on the phylogeography of Rhabdia species, nematodes collected from Bufo species across Honshu, Shikoku, and Kyushu. His work in this area was supported by the prestigious MEX scholarship, highlighting his contributions in the field. His master's degree in biological sciences from the University of Santo Tomas involved an investigation into the parasitic relationships between the endemic freshwater crocodile or the Crocodylus mindorensis and marine fish parasites, which earned him funding from the IUCN SSC Crocodile Specialist Group's Fritz Haschermeyer Veterinary Science and DOST ASRIDIP scholarships. Currently, Assistant Professor Marcaida is an assistant professor at the University of the Philippines, Baguio, where he also serves as the coordinator of the Science Research Center. He is actively involved in various committees, including the Cordillera Studies Committee, the Research Ethics Committee, and the Department Academic Personnel Committee. His research projects reflect a deep commitment to ecological and biodiversity studies. Notably, he's leading a project funded by UP Seeds on urban ecologies in Baguio and serves as a consultant for DOST JSPS funded study on host parasite connections of trematodes in the Cagayan River. His research has led to numerous publications in internationally recognized journals like Parasitology International. As a dedicated member of several professional organizations, including the Association of Systematic Biologists of the Philippines, Assistant Professor Marcaida regularly presents his research at both national and international conferences. His public service efforts include organizing events such as conversations on wildlife and conservation and participating in key regional development meetings on environmental and health research. With his blend of teaching, research, and leadership, Assistant Professor Marcaida continues to make significant contributions to parasitology and environmental conservation in the Philippines and beyond. So, ladies and gentlemen, let us all welcome Dr. Arvin Marcaida.
Thank you very much po for that wonderful introduction, Ma'am Lei. So, can I share my screen already?
Yes po, Doc. Sure po.
Sorry, naka-enable siya.
Okay na po, Doc.
Ayan, all right. Thank you. Can you see my screen, everyone?
Yes po.
Oh, sorry. Sorry. Naka-ano ba? Presenter view or?
Yes po. Okay na po.
Okay na. All right. So, thank you very much for inviting me to share my knowledge and expertise on parasitology. And I'm thrilled to share with you the key findings from my PhD dissertation, which explored the fascinating realm of phylogenetics and, of course, its application in uncovering the cryptic species within nematode population. Now, specifically, my research focused on the phylogeography of Rhabdia species, which is a group of nematode species with a have a cryptic species and infecting Bufo species in Honshu and Kyushu, Japan. Now, to begin with, so I would like to give you a brief introduction on nematodes. So, as Dr. Karin and Dr. Janelle already mentioned, so nematodes are also known as roundworms, are diverse and of course ancient groups of invertebrate animals that have been present on Earth for over a billion years. And they are ubiquitous, found almost in every habitat, from deep sea vents to polar ice, fresh waters, and even soil, until, of course, to the tissue of plants and animals. So there are about 26,000 to 40,000 species of nematodes, both free living. So the free living is 75% of total number of parasites identified. And we have also a parasitic nematodes. And among this, approximately 16,000 to 30,000 parasites of both plants and animals, humans included. Now, this microscopic, inconspicuous, and seemingly unimportant uh, to humans, and therefore attract attentions only to specialists such as us, are vital to ecosystem services such as, of course, nitrogen mineralization in soils or in nutrient cycling, soil food web, or and biomass. Now, a few, however, of course, cause disease of great importance to humans, as well as domestic and wild plants and animals. So with that being said, nematodes are considered biological indicators and agents of course, insects and pests. So here we have a general structure of a nematodes. So they are long cylindrical worms with cuticle with ridges, striations, or annulations. Now, unlike earthworms, they don't have segmentations, and they are usually typically 1 to 5 millimeter in length, but some can grow up to about 15 to 50 centimeters, like the species Ascaris, which are found in pigs or even humans. So here we have, of course, the head part and the tail part, and then we have here the esophagus of the nematode, which is one of the identified characteristics of it. And then we have the green one is the intestine. So nematodes have a complete coelom or intestine or digestive part. And then they have a reproductive part here. So the red one. For male, we have here the male, and then we have here the female. So the yellow ones are the eggs of the female nematodes. And of course, the sperms of the males are this one. And then we have the tail, which have the bursa, spicules, and genital papillae, which are commonly found in male nematodes. Now, what to identify nematodes? So, what are its identifying characteristics? So, we have here the following key characteristics. So, we have different types of nematode based on their mouth parts. So, we have microbivorous, which are nematodes that feed on bacteria in soil, and they are considered free living. While we have a predator nematodes, which have, of course, a teeth and a buccal capsule for attaching itself inside their host. And then we have a plant parasitic nematode, which have a stylet that attach to the stems of or the roots of these plants. Now, each nematodes or species of nematodes have different types of mouths, so used for different purpose or diet. So we have here the lips for the microbivorous, and then we have here the cephalic papillae and amphids for predators. Then we have the lancet for the plant parasitic. So others also have teeth and cutting plates, while for plant parasitics, nematode, they have leaf crowns, or which are used for attachment in plants, or the stems or the roots of the plants. The next identifying characteristics of nematodes are its, of course, as I mentioned, the esophagus or the pharynx. So they have different types of pharynx, and also their reproductive parts, such as the spicules for males, and for females, their vagina in midsection of their body. We have here these types of reproductive parts of the female nematodes arranged in different location or direction. And then, of course, lastly is yung tail part niya, right? So some of the nematodes have bursa or attaching part. Then we also have sensory organs like the setae, the papillae. These are the papillae. If you can see the accessory or sensory organs, and they also have the amphids, phasmids, the labia or the lips, and they also have the skin folding or the lateral skin here. Okay. And then we can also identify nematodes based on their uh, development stages. So here we have different types of development stages of nematodes from their eggs. So each species of nematodes have different types of or different morphology of eggs, which are also used for detection or diagnosis or even identifying for morphological analysis. And then we also have here the different phases or the stages of adult nematodes from juvenile. So we have the L1, L2, L3, L4 until the adult one. So we can use this in identifying nematodes, but we'll see later it's difficult to identify nematodes based on their eggs and of course their developmental stages. We can also identify nematode based on their host and habitat. So some nematode species have direct life cycle which only involves one host, and other nematodes have indirect life cycle which involves several or multiple hosts. So we call this specialist and generalist. So the generalist one includes, of course, or parasitizes different types of hosts in vertebrate hosts, while of course the specialists are only found in certain hosts. So by considering these characteristics, so we can accurately identify nematode species by and understand their ecological and medical significance. So why species identification matters? So species identification is more than just taxonomy or systematics. It's a crucial step in understanding the complex relationship between nematodes and their environment. So firstly, it reveals the evolutionary relationships which help us trace the history of species divergence and coevolution with host or environment through phylogenetic relationship. So it also helps us in distinguishing closely related species within different life histories, host preferences, and ecological roles. Okay. Secondly, it enables us to track infection sources. So identifying reservoir and vector hosts, so we can also determine the transmission pathways, and of course, this will be important for disease management or epidemiological studies or mitigation. Okay. And thirdly, precise identification is vital for treatment development, which is used for tailoring therapies to specific parasite species that optimizes drug efficacy and, of course, minimize the resistance. And lastly, we have the ecological impact. So understanding nematode species help us assess their ecological impact so we can better grasp their roles in ecosystems, nutrient cycling, energy flow, and informing sustainable agriculture and even environmental management. So it is also essential for conservation and environmental monitoring by tracking the nematodes diversity as bioindicators. Now, while accurate identification is crucial, so nematode species identification, of course, faces significant challenges as mentioned by the previous presenters. So these are the following. So we have a small morphological differences in nematodes. So nematodes exhibit subtle variations in shape, sizes, and structures, making visual identification difficult. So we have some nematode groups that have huge cryptic species, which we will discuss later. And we have what we call the morphospecies, where these species of nematodes have a very similar appearance or morphological characteristics. Okay, so as we can see here, this is an example of a Anisakis simplex complex species. So if you can see here, their morphological characteristics are almost similar, and they only vary on circumstances or small details like their length or some accessory or sensory organs, or even in their sizes or shapes. Okay. Now, nematodes have high genetic diversity. So even within the species or population, their genetic variation can be extensive, obscuring clear distinction. So significant genetic variation within and between population. So this is what we call the infra within population and inter-specific variation with among other species or population. So the diversity complicates efforts to identify species accurately based solely on morphology. The next one is we have an overlapping habitats and hosts. So many species co-occur in the same environment, sharing hosts and habitats, and further complicating identification. Now, as I mentioned earlier, so in one host, we can find several parasites, may it be trematodes, nematodes, cestodes, and of course, this includes several species. So it's very difficult to identify based on their habitat or their host. So therefore, there's this may lead to misidentification or misclassification and underestimation of species diversity, or even inadequate understanding of their ecological and pathological roles. Now, to overcome these challenges, so we rely on both traditional and modern. So before the invention or the innovation of the modern techniques such as the molecular, so we mainly use the traditional by using, of course, slide specimens and viewing them in microscopes and looking at its morphological characteristics, drawing them, and of course, in systematics, this cause a several confusion or some revisions or taxonomic revisions or positions or classifications. So now let's discuss the transformation of nematodes identification from traditional to modern. So traditionally, we relied on morphological based approaches, so examining their body shapes, cuticle, ornamentation, mouth, and tail structure. And this requires a intensive and extensive expertise, especially for species with minor morphological differences. Now, fortunately, technological advances have revolutionized nematodes identification. So we have molecular methods like DNA barcoding, phylogenetic analysis, and next generation sequences, which enables enhanced accuracy, higher resolution, and increased efficiency. Okay? And molecular techniques reduce ambiguity, allow detection of cryptic species, and improve identification accuracy, especially in high diversity regions. Okay. Now, as I mentioned earlier, so we have limitations when it comes to traditional methods, especially when it comes to cryptic species. So what are cryptic species? So cryptic species are generally distinct species that are morphologically identical or very similar. So they share similar appearance, and they may also occupy the same or overlapping habitats or hosts, and they may have identical or near identical morphology. Now, the consequences of this cryptic species are misidentification, of course, or misclassification, as I mentioned, underestimation of biodiversity, and even incorrect ecological and evolutionary assumption. Now, detecting this cryptic species require molecular markers via DNA sequencing, phylogenetic analysis, and even integrative taxonomy, which combines both traditional and modern methods. Now, unveiling cryptic species reveals hidden diversity, refining our understanding of the natural world. Now that we've tackled enough information on nematodes, so let's explore about rabias, which is my target taxa, and as a fascinating group of nematodes which have a lot of cryptic species. Now, rabias species belonging to phylum Nematoda, class Rhabditidea, order Rhabditida, and family Rabiaceae. These worms are known for parasitizing amphibians, reptiles, and even mammals. Now, some including humans, but we will be dealing rabies that infect mostly anurine species like frogs and toads and some amphibians. So member of this genus are endoparasites, meaning they live within the body of their host, primarily inhabiting the lungs of this host. That's why they are called lungworms. They also inhabit the soil during their free living stage. Morphologically, rabies species exhibit distinctive features such as smooth, elongated body and a small oral cavity adapted for parasitism. Now, there are 90 nominal species that are widely distributed globally, excluding Antarctica, and they have a less morphological variation. Now, in my study, there are only 9 species in Japan, which I will mention later. Okay, but now let's focus on the hosts of this nematode. So in Japan, okay, we have what we call the Japanese toads that are divided into 2 or 3 species. So the Bufo japonicus and the Bufo japonicus formosus and the Bufo torrenticula. So they are widely distributed across western and eastern Japan, respectively, while having a parapatric distribution, meaning there is a uh, boundary, or there are overlapping boundaries in this region where we can also find the species Bufo torrenticula, which are limited only to mountainous areas of central Honshu. So this is the central Honshu. Then we have the Shikoku and then the Ryukyu. Kyushu, should I say. And of course, there are also instances where they are introduced to some regions or localities. Now that we have discussed its host, let's go back to Rabia species. So these genus have 2 generation life cycle. One is the gonochoristic free living, and the other one is the parasitic hermaphroditic. So when we say hermaphroditic, they have both male and female in one individual, while the gonochoristic free living have separate individual for male and female. Now, the rabies life cycle includes, of course, the egg will be excreted through the feces or the urines of the frogs, and then they will develop in the soil. And then once they have found their host, so nematodes, by the way, find their host or parasitize their hosts through chemotaxis, or the detection of biochemical or organic chemicals released by their host. And then once they detected this, so they can now easily penetrate through skin or oral digestion of their host. Now that you have learned a little background information on Rabia species, so here are some of its species together with their hosts. So we can see here the specialists and the generalist nematodes or parasites. So one, we have the Rabia stockuensis, discovered by Wilkie in 1930. So this is a specialist nematode or Rabia species because they mainly infect only amphibians such as salamanders of Japan. While the Rabies nipponica, which was discovered by or identified by Yamaguchi 1935, as we can see, have several or multiple hosts of different species or belonging to different families of anurans. So we have Bufo or Rhinella, so toads. We have the Rana and also Figervaria, Hyla, so several anuran species. Next is we have another specialist or specialist nematodes, which is the Rabies incerta and then the Rabies cafunata by Wilkie and Sata et al. Respectively. So the Rabies incerta is a specialist one because they mainly infect the Japanese bufo, which is the Bufo japonicus. While the Rabies cafunata was recently discovered in Bufo gargarisans miaconis, which are or can be found in the Ryukyu Islands or the Okinawa region of Japan. So they are very isolated. So maybe this species of Bufo gargarisans, of course, differentiated or speciated from the bufo in mainland Japan. Now, in my study, so what we did was to collect several species of frogs and even salamanders from our collaborators. And we collected their parasites. So here is a slide, a map pinpointing at different localities across mainland Japan where toads were collected. So several species of frogs and even salamanders. So they are marked by following shapes, which corresponds to different hosts. Okay. And sampling involves, of course, catching these toads using net or hands, and we were able to collect 120 toads from different localities, and we dissected all of it, and using, of course, we anesthetized them or sacrifice them for research using chloritone, and then we wash them with 0.9 saline solution. And then we put them in a vial, which includes 90% ethanol, both for morphological and molecular analysis. Now, in morphological method, so we usually mount these parasites using glycerin water for clearing, so we can further see their internal structures, and then we mount them temporarily for using glycerin gel or lactophenol, which includes, of course, some use cotton blue dye, and then we can also store them back to its original storage, like the vial with 90% ethanol. So using both glycerin water or glycerin gel or lactophenol, so this serves as, of course, we can preserve them for long-term storage. And then we measure their morphological characteristics, their measurements, morphometrics, and such. Okay. And then, of course, in traditional method, we also have the morphological or modern method, should I say, the use of the SEM. So before, in systematics and taxonomy, so identification of parasites only includes, of course, using traditional or compound microscopes. But now we have the advanced technology. We now have SEM, so we can further see the minute or the very detailed morphological characteristics of this nematode. So what we did was to prepare them by using freeze drying. So we gradually dehydrated them using ethanols, and then we submerged them in liquid nitrogen and then acetone, and then we freeze dry them for overnight, and then we gold coat them and observe them in SEM. Okay, now let's go to the molecular part. So before mounting the parasites, we first cut a small tissue from its trunk or any parts of its body that is not important for identification, of course, and then place inside a microtube vial, and their DNA was extracted using alkaline lysis by putting 20 to 50 microliter of 0.02 molar sodium hydroxide inside the microtube vial, and then boil under the PCR for 10 seconds with vortexing at the middle. Now, then we replicate this DNA using PCR, of course, under specific thermal condition. And after that, the products were checked using gel electrophoresis with Midori green dye. And then this product were then purified using Promega purification kit and the designated primers. So in my study, we use the design primer of my co-advisor, which is Dr. Nakao Sensei. And then we also use some universal primer, since in nematodes it's difficult to find primers, so we mainly use of course the universal. And then the PCR samples were outsourced to sequencing companies such as PASMAC, and upon receiving the sequences from them, so we then aligned and analyzed them using the Finch TV and the Mega version 10 or 11. Now, in this study, we used mitochondrial DNA COX-1 or CO1 and the ribonucleic DNA to 28S. And yeah. Now, in this study, we were able to identify at least 3 or 4 species. However, only one morphological, only true morphological, and that is the Rabies incerta. So the Rabies incerta was first described by Wilkie, Yamaguchi, and Kuzmin. And this species of nematodes or rabies have a six lips, prominent lateral lips farther from oral opening. So as you can see here, and they have an oral opening or buccal capsule which is wide and round. So this part, the cup-like, and they have a conical tail, wide at base, and narrow in posterior half. Then through morphometry or morphometric analysis, so we compare this species in this study and the ones reported from the Japan and of course in Russia, and we observe a little bit of or a little bit smaller and have shallower buccal capsule. So as we can see here, and all other important characteristics are all coinciding with each other because of what we call the allometric growth. So the allometric growth, of course, in nematodes or parasites are very overlapping, making it difficult to identify using morphometrics. Okay. We also compare the Rabies incerta of this study from different bufo species or clades from the phylogenetic trees, but unfortunately, all measurements are also overlapping. We found no significant variation among them. That's why we rely on the more molecular aspect of this study. So we were able to generate 47 novel sequences of CO1 or COX-1 and 3428S and 718S of Rabies species. And we were also, we also compare it with 20 related species from the DNA Data Bank of Japan, and coming from, of course, several host species like Buffonidae or family Buffonidae, Ranidae, Rhacophoridae, Salamandridae, and Hynobidae. Now, the CO1, 28S, and 18S were, of course, deposited in DDBJ or GenBank as a voucher specimen. And this is important, as Dr. Gakad mentioned in her presentation, in, of course, publication or research. Now, we have here a table of the alignment showing, we'll just focus on the genetic divergence. Now, in analyzing phylogenetic and phylogenetic studies, so in order for us to conclude that a species is a new species, not a cryptic one, so they must have a higher than 2 genetic divergence range. So we have here 54% for 28S, and then we have here 13 to 14 in CO1 or COX-1 in mitochondrial. Now, in 28S, this is mainly used for identifying parasites or nematodes within, between or interspecific population. So we have other species of nematodes. While in COX-1, we mainly use this for identifying within this species or the population, or we can also use this for identifying or revealing cryptic species. Now, this next few slides shows the constructed phylogenetic trees from generated sequences. So I will teach you how to read phylogenetic trees or even read only phylogenetic trees, of course, from journals. So here we use, of course, ML, which is the maximum likelihood and this is common in phylogenetic analysis of 28S or the ribonucleic acid DNA. So this tree just shows us that all our samples, so we have here, so we use this to check if all our samples along with the other family or class of nematodes belong to the same group. So we have here the out group, which is the Circoidea, and we have here another group of nematodes, which is the Strongyloididae and Aloionematidae. Okay, and this just shows us that the relationship among the representative family of Strongyloididae and Aloionematidae, which are all three families belonging to the same Rhabditida, and shows us that all our samples belong to the same family, okay, the Rabiaceae. So we are sure that all our samples now are Rabies species, and this is shown because we have a 100% bootstrap value. Now, if this bootstrap value is lesser than 60%, now this asks a question if they are related or closely related, if not, or distantly related, okay? And then if they are belonging to the same cluster or not, okay? Now in this tree, so we have the maximum likelihood based on partial mitochondrial CO1 regions, and we saw a several clustering or clades in Rabies incerta or Rabies incerta, and this shows us that Rabies incerta consisted of five sub clades. We have clade 1A, 1B, 1C, 1D, 1E, and 1F. Now, this reveals, of course, cryptic species within the species, morphospecies Rabies incerta, and they vary across regions or, of course, the locality of their host. Now, as we can see here in clade 1A, later we have the map. So I'll just explain this one. So clade 1A belongs to Kansai region, and then we have a polytomy, which is still uh, unresolved taxonomic position of this Rabies from the Bufo torrenticula in Nara. So this is still needs further clarification. And then we have clade 1C in Kinki or Kyoto region. And then we have clade 1D in southern and also 1E in northern part of Japan. Now we have a clade 1F, which we think is a new species, which is a cryptic species because we, by true morphological analysis, so we also see it as a Rabies incerta, but their genetic divergence shows it otherwise. Now, we also have a clade 2, which represents a separate species from Rabies incerta, which is the Rabies tokyoensis from salamanders. And we also have a probable cryptic species, which is the Rabies species 1 and Rabies species 1 from a Bufo host. Now, this is interesting because we think that the Rabies tokyoensis nematode or its Rabies species found in salamander host switch from salamander to frogs. Now we have a generalist Rabies species here, which is the Rabies nipponica, which parasitizes several species of anurans like Rhacophoridae, rana, and they are clustered in several clades and found in widespread location. And then we have the outgroup, which is used as a more distant relative of these species of nematodes from Rabies, and they are commonly found in snakes. Now, this slide shows a better illustration of the biogeography, incorporating the phylogenetics and biogeography of each clades of Rabies from this study. So as we can see here, so clade are 1A, 1B, 1C, 1D, and 1E. So the blue one are the Rabies incerta, and they are distributed based on their hosts. And also for Rabies Rabies tokyoensis and Rabies species 1 still needs further clarification or studies. While of course, as we have here the Rabies nipponica as a generalist, so they follow their of course their host, so meaning they are also widely distributed across Japan. Now, this confirms the study from the study of my collaborator Fukutani, Dr. Fukutani from the Kyoto University. So revealing that the hosts now have a genetic variation, and now they plan to, of course, already propose proposing to separate the two subspecies of toads within Japan. So as you can see here, so very distinct yung genetic materials ng Bufo japonicus japonicus and Bufo japonicus formosus. Okay? Now, what are the key findings in this study? So, rabies species is a wide host range. So, they parasitize amphibians and reptiles of various orders and families. So, ranging from specialists to relative generalists. And they could be tracking host resources instead of phylogeny. So, rabies species shows significant genetic variation, suggesting cryptic species. And as I mentioned earlier, so they have broad host range and resource tracking rather than strict phylogenetic alignment or coevolution between within their hosts. Now, there are also overlapping or sharing of habitat, of course, from terrestrial versus semi-aquatic frogs. So frogs have different microhabitats. We have the arboreal, we have the terrestrial, and then we have the semi-aquatic to aquatic frogs. Now, physiological aspects such as epidermal properties or defense mechanisms also play a crucial role in, of course, transmission. So factors, and also evolutionary ability such as yung host switching nila, so facilitate cross transmission. Now, rabies incerta exhibited a higher level of variation in this gene due to the following, such as the differences in the host segregation or isolation, because as we can see from the map of the hosts of Bufo japonicus, so they are very separated, western and eastern, because of allopatric speciation or yung tinatawag nating pag-separate nila by physical factors such as formation of mountains or even lakes or forming of islands. So it reduces the genetic flow and resulting to diversification or speciation of these species. Okay. Now, the key takeaways in this presentation is, of course, the highlighting the roles of molecular data in accurately identifying nematode species, especially if you have cryptic species within the family group. Okay. Now, implications of this is, of course, the biodiversity assessment, conservation, and ecological research, and even medical research. Okay. Now, I would like to thank, of course, my research team from the lab of Dr. Urabe Misako, and of course, University of Santo Tomas research team. And yeah, thank you very much for the time and of course, listening to my presentation.
Thank you very much po, Doc Arvin, for that presentation. Ang cool po ng mga present nyo na data, no? Tsaka yung phylogenetic tree, actually, that was so informative. So, sige po, we will start the discussion po from these questions dun sa ating Q&A box as well as dun sa ating YouTube live. So, ayan, Doc, yung first question po would be from Sid MJ Ramos. Good morning, Doc. What software did you use in constructing a phylogenetic tree? Yung Mega po ba or yung Beast? Thank you po.
Very good questions. Thank you for that. I usually use the Mega since I'm not really used to coding or forming HTML, because some of the software or program for phylogenetic analysis include yung coding nga, which is very difficult. But if you have the knowledge and even the resources or references, yeah, you can use that also. For Mega, it's easier to use because of its, of course, yung pwede mo siyang maklik-click lang, and then mas madali siyang gamitin, very friendly. And yeah, that's all.
And thank you po. And the next one would be, Doc, what are the common species of nematodes affecting the toads or frogs here in the Philippines? And how can they affect the freshwater fish species, especially those living in pond aquaculture?
Very good questions. Thank you for that. So I think one of the families that we can, or families of nematodes that we can commonly found in or find in frogs, and some might infect, of course, fishes, or we have the strongyloides. Okay. We have, I think mostly several families from Rhabditida. Okay, so I think that's most of the common ones that we can find in frogs or even some fishes. Okay, thank you very much for that.
Sige po. And next po would be, what are the significance of nematodes in ecological and medical aspects po?
Okay, thank you for that question. So, as I mentioned earlier, in ecological, so we can use it for example, environmental health. So they serve as a bioindicator or biomarkers. So, some nematodes are used for, of course, detecting heavy metals, or even pol- uh, nutrient cycling, or in study of biomass. And for medical aspects, so there are some family or groups of species of nematodes that can, of course, cause diseases. We have yung angiostrongylos, or kaya naman yung mga ano tawag dito? As I mentioned, yung sa pig or sa pork, nematodes, I forgot the I mentioned earlier. So we can use them for, of course, biological indicators, of course, for the host fitness or health, okay, and of course detecting their diet also. So we can determine the life cycle of these nematodes, and then we can also determine their, of course, diet. Okay.
Then next question po would be, what are the primary challenges in identifying nematode species, especially those when that share similar habitats and hosts?
Okay, very good questions. Thank you. As I mentioned earlier, yung pinaka-main challenges in identifying nematodes are yung cryptic species, kasi maraming cryptic species within nematode families or class or order. And very tedious identify especially morphological characteristics ng nematodes, dahil it requires kailangan maano ka sa detail. So even the smallest structure of these nematodes, as I mentioned, yung mga amphids, phasmids, so we have to really check it out. So without fortunately, with using SEM, so we can now see these minute characteristics, and we can identify them up to species level. But if there are some cryptic species such as the Rabies incerta, I think it requires a lot of time and very tedious to really see the difference or the variations within their morphological characteristics.
Alright, sige po. Moving on, may mga may mga instances po ba that rabies species have infected fishes? And are there any distinct symptoms related to that? Are there any distinct symptoms in relation to infection sa fishes?
That's a very good question. So, before, during my undergrad thesis, so we were able to, conducted a study of fish parasitology in one of the 7 lakes of San Pablo. So, unfortunately, we weren't able to find as much nematodes as from the vertebrates, like frogs or even mammals. So I think only a few recorded species of nematodes have been identified or studied in I think Philippines, but I'm not sure which family they belong to or species. So of course, as mentioned by Dr. Gakad in her presentation, so there are also change in behavior, of course, swimming patterns and such. In frogs or amphibians, the Rabies incerta cause pneumonia, and even to some vertebrate hosts such as, of course, mammals, but they are asymptomatic at first. But if they are immune compromised, and then these parasites may, of course, cause greater damage to the lungs and will affect their fitness or host fitness.
Okay po, that will be for our Q&A box. Then moving on sa atin pong mga YouTube participants po, may question po from Miss Canessa. Have you tried to extract DNA from fish nematodes larva for small larva like anisakis? Will it be more useful to extract from whole worm or from just a part of the worm like the middle portion?
That's a good question. So, however, I haven't extracted or conducted yet a nematode study on fishes. Actually, I also plan to do that here in Philippines now that I'm establishing a career. So, it's one of my plan actually, along with my collaborators such as Dr. Jonathan Briones, Dr. Gakad, and Dr. Karin. So we also plan to, I hope in the near future, we plan to conduct nematode survey on fish in several freshwater areas in the Philippines.
Nice one, Doc. And another question would be, what software, Doc, do you recommend for computing the genetic variation to detect cryptic species?
That's a good question. For me, I only use Mega. However, I think there are several computing softwares such as R or Bees even, and I think there are several software that you can try. But it's easier, as I mentioned earlier, it's easier to use the Mega because of this friendly user interface. So yeah, you can try several. Thank you very much.
Ayan, and to cap this discussion, Doc, last question would be, in your on your studies on nematodes, how does it contribute to the treatment development in relation to its prevalence in the country?
That's also a very good question. Thank you for that. I think, as I mentioned, so identifying is really the first step in, of course, for mitigation or disease management. Of course, this is also applicable to other parasites or viruses. So, of course, your first step is to identify the species. So, for example, in one of the paper I read on anisakis, so there are some cryptic species that they were able to identify, and this is clearly separate from the regions such as the anisakis in Japan and the anisakis in Europe. So I think some of these species may have a different disease that can inflict in their hosts. And of course, with this, we can tailor the medicine or the uh, treatment for this parasites. So in treatment, so we already have, of course, several medicines in veterinary and agriculture. So we have deworming, and we use this for livestock. So I think we can start by identifying first which parasites are found in our, for example, livestock or ruminants or yung mga farm animals natin if we're going to use this for agriculture purposes.
Okay, sige po. Ayan, maraming salamat po, Doc Arvin, for giving us your time. Especially, alam namin na napaka-busy niyo din po sa inyo pong sa work. Ayan. So,
Alright.
ayan, maraming salamat po, Doc Arvin, and we hope to see you po sa atin pong mga future FishKwela activities. Ayan. Sige po. That was Doc Arvin Marcaida sa atin pong third topic for this FishKwela activity. And maraming salamat po, and yung susunod po ng mga lectures will start at 1 p.m. po mamaya. Yan. So magbibigay po kami siguro ng konting recap sa mga nangyari kanina before we proceed sa next topic. So, ayan po, that ends our morning session, and kita-kits po tayo at 1 p.m. po dito pa rin sa atin pong Zoom platform. So, ayan, please don't forget po to Ayan, to register po doon po sa ating attendance form. And later on, we will also be presenting to we will also show yung mga ano po yung mga post test links, yung evaluation links which you have to complete po para sa atin po CPD requirements. Ayan, sinend na po din ni Ma'am Antoinette sa atin pong chat box yung link for the attendance for your reference. Ayan, and kung wala na pong questions, kita-kits po tayo mamayang hapon. Ayan, yan po sa ating morning session, PM session. Maraming salamat po. Good afternoon. Hello po, good afternoon. Hi po sa mga nandito pa po sa Zoom. And mag-start po tayo in 8 minutes po. May mga ano lang po siguro, mga, ayan sige, yung mga babalik po sana na ano na participants na umalis kanina. Sige po. Hintayin lang po natin saglit. Then, okay. 7 minutes. Thank you po. Hi, good afternoon. Ayan, may mga participants na tayo na pumapasok na po sa atin pong Zoom platform. Ayan, mag-start na po tayo at exactly 1 p.m. No? So, ayun po, natanong ko po kanina, as a refresher, ano pong takeaways niyo sa tatlong presentation na natapos natin kanina? So, can you comment them down sa ano po, sa atin pong chat box para po makapag-start tayo no ng parang ano lang pang-refresh lang, konting ano ba, konting exercise, mental exercise para sa ating afternoon session. And sige nga po. From our attendees, may 110 attendees na po tayo. And ready na actually ang ating. Okay, sige. Anyone from our participants, ano pong takeaways nyo? Ano pong naalala nyo dun sa presentations nila Doc Harin, Doc Janelle, and Doc Arvin? Wala? Anyone? Sige, babalikan natin yan. Babalikan natin yan maya-maya. No? Sige, i-ano niyo lang po, i-comment niyo lang po dyan. And also sa atin pong YouTube participants, pwede pa rin po kayong mag-participate sa atin pong discussion. Okay, sige po. I-ano lang po namin, i-forward na lang po dito ng admin po yung mga yung mga comments ninyo. Sige. So, to start po yung yung presentation for our next speaker, papakilala ko lang po muna siya, no. Our next speaker po is Dr. Jonathan Carlo A. Briones. So, Doc Briones is currently a full-time faculty at the College of Fisheries Cardona campus sa University of Rizal System. So, he finished his Doctor of Philosophy, major in Biological Sciences cum laude at the University of Santo Tomas in Manila. He also finished his Master of Science major in Biological Science at the same university. So, sige po, he has also been an associate professorial lecturer at the De La Salle College of St. Benilde, Antipolo, and assistant professor, instructor, lecturer, and a teaching assistant at the University of Santo Tomas, Manila, Department of Biological Sciences, College of Science. So, ayan po, he has co-authored a book on Earth and Life Sciences, Knowing and Understanding Nature, with Papa and Tordesillas in 2016. He has also presented various publications po in international publications, such as his study on the gut helminth assemblages of Glossogobius aureus in Taal Lake, Philippines, and low levels of Trichinella antibodies detected in domestic pigs at selected slaughterhouses with farm-based exposure assessment in Bulacan, Philippines. Nutrient loadings and deforestation decrease benthic macroinvertebrate invertebrate diversity in an urbanized tropical stream system. Stream benthic macroinvertebrate assemblages reveal the importance of a recently established freshwater protected area in a tropical watershed. Then the curious case of the endemic freshwater crocodile, the Crocodylus mindorensis, as incidental host of marine fish Acanthocephalan, fish diversity and trophic interactions in Lake Sampaloc, among others. So he has also published in national publications such as his study on the review of the biodiversity and limno-ecological studies on Taal Lake updates from the last 10 years. Then invasiveness risk assessment of non-native freshwater fish species for Lake Taal, Philippines, and Aeromonas veronii and Plashamonas shigaloides isolated from Glossogobius aureus in Lake Sampaloc, Laguna, Philippines. So those are among his national publications. So he has also joined trainings and seminars po internationally and locally, and has presented his various works in this symposium. So, di ko na po siguro papatagalin na-excite na din kami as well as you, no, para marinig itong presentation ni Dr. Jonathan Carlo A. Briones. So, here's Dr. Briones for his presentation po. Hang on po, may konting difficulty lang po on the technical aspect. Wait lang po.
Magandang araw sa ating lahat at maraming salamat sa pamunuan ng NFRDI para sa opportunity for us to be able to talk with all of you today. To begin, we would like to ask you a question. Sa mga mangingisda sa inyong mga komunidad, are they fishing mainly for subsistence or for income? This is an important question because now we know that inland fisheries is an important and integral aspect of achieving the Sustainable Development Goals that was set forth by the United Nations. Dito, we can see how each Sustainable Development Goal can be addressed by or positively influences development in inland fishery industry. However, Philippine fisheries is at risk at the moment because of many different factors, the biggest of which would be invasive species, as well as the onset of climate change and how it affects both capture fisheries and aquaculture. Now, we need to understand that we have so much technology and we can actually link this towards capture fisheries, aquaculture, and find potential implications on how it can be related to environmental factors and even biotic factors. That's why I would like to share with you today the importance of big data for fish biology and fisheries in the Philippine context. I'm Jonathan Carlo A. Briones, and I am a faculty of the College of Fisheries of the University of Rizal System Cardona campus. You may be wondering, what is big data? Well, big data, in its usual definition, are large amounts of data that are passively collected from digital interactions. So a perfect example would be when you click enable cookies when you visit different websites. When you do that, what happens is that you allow these people to obtain a subset of your information, which they then share to certain advertisers, which provide you with ad-focused advertisements. However, when I talk about big data, I'm merely mentioning the scale or volume of the dataset. As for our working definition, this dataset may come from a variety of sources and might be of different types. And of course, some of them may have some level of uncertainty, which we might need to validate or check much later. Later on, I'll show you an example of where we can find such big data and how we can use them for research and other purposes. Now, going back to our main topic, which is the importance of big data, it's of value to us because it tells us a lot about long-term trends. Trends such as interactions between parasites and hosts, which you've seen in the previous talks. It also shows us how these parasites and hosts, which are all biotic components, might be impacted by environmental changes, which then impacts the economics of fisheries as well as our human health in the long term. So we have, on one aspect, big data on fisheries, and on the other side, big data on fish parasites. And we hope that after this talk, we will be able to give you an understanding and an appreciation of how we can link these concepts of big data for us to find trends and research questions that may not necessarily need a lot of finances, but rather more on your analysis and efforts. So let's begin. Unahin muna natin ang fisheries big data. With fisheries big data, we actually utilize this as a solid foundation for informed management strategies. Back in the 90s, there used to be the conventional fisheries management, wherein we only focus on a target stock. We focus on kung paano natin siyang paparami uli, mape-preserve, mako-conserve. But now, going towards the 2000s, we know that even now, with NFRDI and BIFAR, they endorse the ecosystem approach of fisheries management, wherein rather than just looking at one subset, we're actually looking at the interrelations, the ecology of it all across different components of the ecosystem. And this is important. What we thought of as not important components of the aquatic ecosystem, such as macro or micro invertebrates, are actually an aspect in a very long chain that affects our target fish stocks. Now, talking about different papers that discuss long-term fisheries with climate change, here we can see a paper by Jenner and his colleagues in the journal Global Change Biology, wherein they look at body size dependent responses of a marine fish assemblage versus climate change. And it's not in any way good. Why? Because climate change in the marine aspect seems to show that fish tend to become smaller and catch becoming much lesser. Why is this happening? In this particular paper, they were able to see that the length and the weight of the fish catch for a long period of time, they have a very large data set back from the 1910s up to the 2010s. Fish catch in terms of length and weight per individual fish is becoming smaller and smaller, while catch per unit effort has also been lessening to a certain degree across different species. Now, if we investigate it even further, we can see that looking at ecosystem-based fisheries management is an important step for us to forestall and become resilient against climate change-driven collapse. In this paper published in Nature Communications, we can see and read that if there is no intervention made with regards to fisheries management, there definitely is a trend of higher risk of decline in fish catch. And unfortunately, this projection of change in fish catch is species specific. Some fish species can thrive or become more resilient compared to others. And unfortunately, the ones that are more resilient are usually the non-native fish species. So that gives us an understanding that conserving native fish species and managing them for long term should be an important target if we are to decide on an ecosystem-based fisheries management. Here, we would like to give you one example from our own work, which we published in PLOS ONE. Here we studied a native fish goby from Lake Biwa, Japan. Lake Biwa is the largest and most ancient lake in Japan, wherein the Japanese researchers were able to obtain a very long data set of 40 years. What did they do? For 40 years, and even up to present, They obtained fish samples of the same species and they retained them as museum samples across many, many years. The sampling regime is the same, and because of this, they're able to ask many important questions about what's happening in the lake and relate it to the fish, taking information from the fish in the particular year that it was sampled. For example, in Lake Biwa, there was a trend that back in the 1960s, total phosphorus, which is an aspect of nutrient increase, was actually increasing from the 1960s going towards the 1980s. And this is in line with the Industrial Revolution, diba? So, and from then on, the Japanese government enacted certain laws that actually lessened total phosphate use across different usage, whether it's in agriculture industry or in households, such as the ban on the use of phosphate-based detergents, which actually, shown here, really decreased total phosphorus in the waters of Lake Biwa. During this time, we were able to see that the native goby fish, which we call isaza, actually changed its diet between the 1960s to 1980s, and then after the 1980s to the 2000s. We're able to see that the goby fish initially was first eating a plankton called Daphnia from the 1960s to 1980s. But starting in the 1980s, there was a trend happening wherein it started to eat more of the benthic gamarid, which is an amphiphod. We can see this both in number composition of the diet and as well as the weight composition of their food items. So here, we can see and relate how these changes in diet can actually be related to the changes that are happening in the lake itself, which is related to policy changes and also climate change driven impacts. Now, with regards to fisheries big data, I want us to appreciate what we have here in the Philippines. For example, we have tawilis, which is endemic to Lake Taal. For Lake Taal, the catch fisheries of tawilis and also biyak feed and support the fisheries communities around Lake Taal. However, we know that tawilis has been declared decreasing in its current population trend and is now endangered. It was first assessed back in 2017 by Doc Muji themselves and components of NFRDI and various BFAR representatives. So now, I think it's already improving and it requires a certain level of reassessment. How can we use big data to be able to assess tawilis? A possibility is for us to explore long-term seasonal variations of tawilis from Lake Taal and to be able to assess implications for conservation management that is already being enacted. And this is important. Una, dahil masarap ang tawilis. And of course, from the biologist aspect, tawilis is one of those species that are of high conservation value. How can we do these possibilities in our own backyard without the need for finances, immediate finances? The easiest thing that we can do is obtain data freely online. We have to tell you that there is a need for us to appreciate the work of the Philippine Statistics Authority together with various regulatory agencies from the Philippines. They have a variety of OpenStat data that is accessible, such as demographic and social statistics, economic statistics, and environmental and multi-domain statistics. So going back to tawilis, how then can we look at big data related to tawilis here in the Philippines? One way that we can do this is to look at fisheries data, agriculture, forestry, and fisheries. Clicking on that, we choose fisheries. And here, I'm just highlighting some of the data sets that you can see, but we actually are able to obtain inland municipal fisheries, volume of production by geolocation, species, year, and quarter. Now, this is just one example of available data sets that we can obtain. From this particular data set, we can see that there's actually a 22-year-old data set from 2002 to 2023. However, there are certain limitations in the data set itself. One example is that the data set available online freely only has quarterly resolution. So it's clustered as well, with no distinction between municipal landing areas. So how are we able to discern why tawilis is observable from this dataset? Well, the dataset itself only provides cluster data. But since we know that tawilis is only available in Lake Taal, we can now create certain inferences that is particular to Lake Taal. So in this dataset, each data point now becomes quarterly volume production per metric ton. So that is the metadata available for the dataset itself. So what is the news that we know? We know that tawilis capture fisheries has declined throughout the years. So back in 2002, it was actually low. Going back to 2007, 2008, and 2009, there was high production. And from this point, it has actually been declining. If we were to analyze this data set of tawilis volume production, we try to look into quarterly volume production and related to yearly volume production, we can actually see that quarter 2 of tawilis volume production has a distinct, strong relationship to total yearly production. Here we can see an R2 of 0.687, followed by quarter 1 and quarter 3, with the least at quarter 4. What does that mean? At present, in Lake Taal, ang tawilis merong moratorium, which is around quarter 1 and quarter 2. And here we can see that the data set from the last 22 years is highly related to the production of what happens between quarter 1 and quarter 2. Thankfully, the moratorium was set in Taal to be able to help increase the production of tawilis, knowing that it's biology that they actually breed during that period. So here, looking at big data for tawilis, we can affirm that certain actions and policies, such as the moratorium that was set, is actually related to long-term changes, long-term positive changes, as we may see here. So that's one way that we can look at fisheries data, big data, how we can see information on a global scene, as well as how we can utilize it in our own country. Now that we've discussed big data on fisheries, I hope you were able to appreciate the various global studies that we showed you and how we can apply that to local information and data sets that we can retrieve for the Philippines. This time, let's talk about big data on fish parasites. We know that fish parasites have been documented for a very long time. One very important parasite would be nematodes, which was also discussed during this FishKwela session. There's a meta-analysis study on fish nematodes that studied its global abundance, particularly of Anisakis and Pseudoterranova. These meta-analyses are very important and valuable for looking into biogeography studies on the presence of these parasites in fish. Now, in this particular study, they used a system to be able to check the records through the Web of Science, wherein they screened at least 1,300 titles and certain abstracts and full texts, from which they were able to check Anisakis from 526 studies, as well as Pseudoterranova from 229 studies. So in this particular work, which is very extensive, highly cited study, they were able to see that Pseudoterranova did not really increase much over the decades in terms of intensity and abundance, but Anisakis itself has increased in a big way in terms of abundance and in various places all over the globe. So that's one way we can use parasites as a means to study their presence. And of course, we know that Anisakis itself does not only affect fish, but also humans as well. Another study that we can appreciate on big data for parasites would be using them as biological tags for fish stock delineation and movement patterns. Sa ating mga fisheries students na nandito ngayon and those who are fisheries professionals, we know how difficult it is to be able to determine the source of a fish stock, especially in marine ecosystems. One challenge that we have is that the biological tag or the tag itself should have some form of longevity, or we need to be able to understand its source and why it must be present or absent, or how it can actually change through time. Some people use fish otholates as a fish stock tag, while some utilize some form of traceable chemical. But parasites themselves, specifically helminths, can be a particular biological tag. In this particular study that was published in the journal called Parasitology, they were able to see how a particular parasite or different sets of parasites can be a valuable stock marker for fisheries in Australasia, East Asia, and the Pacific Islands. Why is that? They know that in certain areas, certain parasites are present. So for example, if we utilize this graph right here, we can see that across most of them, the parasite abundance that is present in black is fairly common across all areas. However, there are certain areas that it is very delimited. For example, in this area right here, parasites are almost not present. And there are certain areas, when we look at other parasites represented by the graph itself, for example, in this site, site 3, only the parasite designated by the black column is present, as well in number 8, this site in number 8. So we can potentially utilize these helminth parasites as biological tags to know where these fish stocks were actually retrieved. Another value for parasites would be, as we utilize them as natural history collections and understanding these natural history collections. Now, one way that we can do this is to analyze museum specimens that have been preserved in fluids. As long as they have been preserved and fixed in a particular fluid, for example, in ethanol or in formalin, they can be valuable resources as well for ecological and biogeographical studies. Now, in this study, published in Ecology and Evolution, which is also highly cited, we can see here that there's a variety of natural history collections that are present even back in the 1900s. So, hundreds of years of available data for natural history collections. Now, for parasites themselves, there's a very small aspect of historical data that is available, and that can be utilized for an appreciable level of meta-analysis. Now, investigating this now, we can see that certain meta-regression estimates, we can study a variety of parasites that can be obtained from fluid preserved fish specimens found in museums or in natural history collections. We know that certain parasites can be harder or can be softer, so we have to understand that particular effect in their detection rate. For example, nematodes might be easier to detect, especially acanthocephalans, because they're much harder, as compared to trematodes or cestodes, because they're much softer. So these are certain considerations that we can see. Also, there are certain limitations that we need to understand. Once we are able to obtain parasites from museum specimens of fish, we have to understand that it might be difficult to identify them to species level. But if our questions are ecological in nature, we might not need to identify them to species level at all, only to a certain level of ecological importance. And of course, we know very well that parasites impact the survival and recruitment of fish populations. We know how much parasites affect our fish stocks. That's why we have a separate subject for them in fisheries, which is fish health management. We know it affects aquaculture as well. So there are a variety of highly published studies, especially on globally partaken fish food products, especially on salmon, wherein parasites really impact these fish stocks. And we know that by utilizing anti-parasitic treatments, it improves survival rates of our fish stocks, as well as parasitism acquired early in life, especially in migration, can reduce survival rates. Now that we know that parasites do affect fisheries, it's also justified for us to ask, how then has fisheries affected parasite communities? This study by Wood and Lafferty, published in Parasitology, shows that parasite diversity, meaning the number and species richness of parasites, is much lower in hosts from fish areas, and larger hosts can harbor more parasites. But the species richness will be different based on whether the area has been highly fished or not. Not only that, but it also depends on the life cycle patterns of the parasites. When the parasites have a direct life cycle, so for example, certain nematodes, the biodiversity effect on these nematodes is much less as compared to certain parasites that require additional intermediate hosts. In this case, biodiversity loss or the decrease of biodiversity in fished areas is much less for parasites that require indirect life cycles compared to those that require direct life cycles. Now, with this, we want to be able to show you how you can utilize fish parasite big data because it's already available. And such software is what we call FishPest. FishPest is a software that utilizes a large data set that was taken from almost 11,800 fish parasite species. And these have already been published in well-reputed journals and have been highly cited. Fish pest and the dataset from which it was retrieved and is also updated offer insights into long-term species-area relationships and host specificity. So, such big data on fish pest, which is an acronym for Fish Parasite Ecology Software Tool, allows for a variety of things, mainly three aspects. Parasite niche modeler, wherein we can model a potential host range of a parasite. Parasite co-occurrence modeler, looking into the co-occurrence of different species, and as well as a potential parasite list generator. Now, how can we utilize this in the Philippine setting? In the Philippines, we have actually been a pioneer site for fish parasite research in tropical Asia. We have a lot of well-known parasitologists, one of which is the national scientist Carmen Velasquez, who studied a lot on the digenetic trematodes of Philippine fishes. This is followed by the academician Eduardo Salcedo, who followed suit on Dr. Velasquez's work. And Dr. Salcedo was able to publish many things, such as the parasites of Philippine vertebrates as an index catalog, specifically on the type specimens of these works. And of course, there are the works of the FAO-UN, together with Lumanlan Mayo and Arthur, which lists the parasites of fishes in the Philippines. So, with regards to fish parasite research, the Philippines is a rich hub that you should still explore. Going back to fish pest, how then can we utilize this of what we know now in the Philippines? Now, let's go to the actual website. Here, we have the website of fish pest, wherein you can check for different genera of various helminths. Here, for example, we are checking Botricephalus achelognati, an invasive tapeworm. We have a variety of options that we can explore, but you want to know how this one can affect a specific fish family. For example, gobiids. Where? In the locality of course, the Philippine Islands. And here, let's say we want to explore only freshwater fish species. And from here, we can create our model output, looking into potential compatible hosts in the Philippines. And with that, we can see an output of the known host set of this invasive Asian fish tapeworm, and of course, to which certain species it can actually also infect. Certain ecological parameters that we can also use for our studies is also shown here. And it also shows likely compatible hosts that are not listed in the known fish species list to which fish pest is currently loaded. Okay. With that, we know that the invasive Asian fish tapeworm is a very invasive fish species that is of high aquaculture value, because in places where it is present, given the right conditions, it will actually destroy the available fish stocks. Looking at the modeled compatibility of this Botricephalus achelognati, which is now updated at Schizocotyle achelognati, we look at one example in the output of fish pest, which is its potential compatibility with biak, Glossogobius aureus. Looking at these variety of parameters, we can see that Glossogobius aureus is within the niche of Schizocotyle achilognathi. So here, and here, we know that the growth parameters of Glossogobius aureus is prime niche for this particular invasive parasite. And this is actually what we found from the recent publication of one of our students, who is now Dr. Vanessa Vargas Martinez, wherein she was able to find Botricephalus achilognati, known now as Schizocotyle achilognati, from Glossogobius aureus from Lake Taal. So with this, just by using FishPest, we're able to see and find potential compatible hosts and see it live if it's actually present. So hopefully, utilizing fish parasite big data, we can use FishPest as a software that can help us with certain investigations that we want to know, particular to your own region or your own research area of interest. And with that, we would like to end this short talk by focusing on these various points to consider, knowing that big data on fisheries, fish biology, and fish parasites really do reveal significant trends in economics, ecology, and human health as well. When we utilize these various opportunities, we can enable ourselves to model certain parasite niches, investigate potential host-parasite interactions, and also look into possibilities into a variety of host-parasite interactions. And lastly, we can explore how the continued use and research of big data may be of value for you, not just for research, but as well for policy advocacies and management strategies that you might use in your own particular area of work. And with that, maraming salamat po. And if you have any questions, please feel free to ask, or you can email me in the email available here. Maraming salamat.
Alright, that was Doc JC.
Magandang
Ayan. Hi po, Doc JC. Good afternoon po.
good afternoon.
Alright, sige po. We'll start the discussion po sir sa regarding sa yung presentation. So currently po yun dun sa big data database, that was a database, di ba? Yes po. Currently po yung database po ba is stable na siya, like nagagamit na siya of institutions like research institutions, SUCs.
So far, ma'am, actually, the data sets that are available, for example, yung sa fish parasite data, it's integrated into the FishPest as a database para pag gumagawa siya ng model. Pero I think in-update nila yun back in 2015. I'm not sure if they're going to update that further now. With regards naman to big data on fisheries mismo, that's something that we can all explore. One way that we can do that, simplest one that I showed you, is to just look at the Philippine Statistics Office. Pero many people will say na there's a level of uncertainty eh. Some people say na, huwag kayo magtiwala dyan, which I don't really understand why. It's not PSA who gets the data, but actually it's the different regulatory agencies who bring the data to PSA that they just co-organize. If you want a higher resolution data set, baka mas importante if you go directly to the monitoring agency source. So for example, right now, nandito po ako sa BIFAR and FITC sa Tanay, and the topic that we just showed you is actually our current research now. Long-term fisheries changes in Laguna de Bay. So we're coordinating with BIFAR Tanay, LLDA, Pag-asa, and pinagbabangga-bangga namin yung mga data set for Laguna de Bay fisheries.
Well, that's interesting to know, no, Doc. Yung database po ba, may mga may mga partner agencies kayo surely na naging partner na din para ma-develop further yung ano na yun, yung database na yun. And kung sino po kayang mga ano na po, national government agencies na po, aside from BIFAR or dito sa NFRDI siguro or sa SUCs ang nakagamit na po nun?
Okay. Actually, I'm not sure. This is just one. The reason why I'm doing this right now is because it's a type of research na does not require finances. So kung may computer ka, you just and you can talk to these people, you can request using yung Freedom of Information Act, magagawa mo siya. So it's just a means for me since to be able to do research without the initial finances. So the data set itself is available only from the monitoring agencies themselves. But one thing that we did, we first initiated institutional MOUs, so Memorandum of Understanding. Nung napirmahan na yon, then we requested for a data sharing MOA, Memorandum of Agreement. So we were able to obtain high resolution access, the highest resolution access na kaya nilang ibigay. Kasi sometimes the data itself is not high in resolution. Kung ano yung pinakamataas, pwede nilang ibigay. So that's one way that we're doing it right now. With regards to the fish parasite data set, it's the brainchild of sila Lafertier. Sila yung, if you visit the website, they're well-known parasite ecologists. And I'm not sure if they're updating that now, but you can actually access it as free data. I think it's open data. You just need to find the link for them to download it. And so available siya fully online.
Yeah, actually, sir, medyo, actually, alam ko marami makaka-appreciate, lalo na yung mga nandito sa, ano na sa atin pong webinar, lalo na yung mga nasa fisheries offices. So with that, sir, may mga ano po ba, may mga programs na po ba or mga projects na po ba wherein naging malaking ano part yung database, yung big data on fisheries po?
For me, I think one thing that we need to give appreciation to is yung ginagawa ng NFRDI together with sila Doc Mutya sa NSAP, di ba, sa National Stock Assessment Program. And the main work is all over the Philippines. You even have a software, you have a mobile app na that accompanies it. So makikita mo siya real time, di ba? But at the same time, si NSAP right now is limited mostly to marine areas. So there's not much on the National Stock Assessment Program related to freshwater. So that's something that we're trying to explore right now. So to the students who are here right now, especially if you're near lakes and rivers, water basins, that might be something that you might want to explore. Kausapin niyo ang NFRDI that you want to work with them. Pag-aralan niyo yung protocol ng NSAP and then sundin niyo siya and then try to follow it suit dun sa area of inland fisheries area na meron kayo.
Yeah, actually, I agree with that, Doc. Kasi lalo na yung mga nagtithesis, ano po, mga ano po yan, marami talaga silang makukuhang data dyan. Sige po, so far wala po tayo masyadong questions pa from our Q&A box, but siguro manghihingi kami ng questions from our YouTube live. From YouTube live, Sir Joseph asked po, "Good afternoon po, Sir Jonathan. Thank you for a very informative talk. Just want to know po, sir, if there are any established online protocols for collecting data on various fish parasites."
Okay, so that's a good question. For me, one way for you to quantify them, there are a lot of papers on quantifying fish parasites. One good place that you can go to is look for Quantitative Parasitology 3.0. So it's a website, and they show you the protocols that you need to be able to obtain values of intensity, abundance, how to obtain them, what data set that you need. So mas mapapadali yung buhay ninyo. Ang problem kasi sa parasites, as you can show, as was shown in the other studies, is that it's not like normal data sets. Some will argue that they have a negative binomial distribution. So kuwari, merong 100 na potential hosts. nagbukas ka ng 100, 2 or 3 lang dyan ang meron, tapos yung 2 or 3 na infected is highly infected. So that means they're not analyzed in a similar manner na usual parametric testing. So those are things that you need to explore with regards to that. Kung paano naman in terms of isolating, ang problem with parasites is that it's taxon specific. So kanina nakita yung si Doc Marcaida talked about nematodes. Si Doc Gakad talked about trematodes. So those are two very different systems. They require different ways to be able to isolate them, and isolate and fix them and identify them. So iba-iba yung processes nila. So it's taxon heavy. So ang preference dyan, para hindi ka ma-overwhelm, pumili ka muna ng isa na gusto mong mahalin. So siya ka na lang mag-venture out. So that's one way that you can do that.
Ayun, tama naman yun, Dok. Sige po. And balikan ko po ng konti no kanina sa mga naging presentations nila Doc Janelle. May mga presentations po kasi doon na parang relevant questions po doon sa presentation ninyo. So may nagtanong po kasi doon sa determination ng parasites, kapag na-identify po nila sa certain fishery species, syempre nagre-raise ng issues kasi na-alarm yung tao, the public. And naging basis po ba kaya yung mga database na yon para doon sa sa paglabas ng mga measures po on what the consumer or the public should do or in order to address yung mga ganun pong concerns?
Okay. So medyo bias yung magiging sagot ko kasi parasitologist ako. And I'm not a medical technologist or I'm not from the medical field. Ang parasites kasi, for us, yung mga helminthologists, many parasites are present in fish who only affect fish and don't really affect humans. Diba? And most of them are not found in areas that humans actually eat. So they're usually found in the innards, laman loob. Madalas hindi naman sila found in the fish flesh. And there's an actual call now to preserve parasites in general. Some parasites in general. Of course, yung mga parasites of human medical importance, those need to be eradicated. But those parasites na are normally found should have some semblance of conservation. Not because of them, but because by using them as a conservation tool, we're actually conserving the environment. So if you remember yung aspect kanina ng discussion, we're always asking what do parasites do to fisheries? But then there's a justified question, what does fisheries naman do to parasites? So extensive fisheries actually decrease species richness in parasites. And that's something that we need to understand. So parasites are here to stay. They're not all of them, a very small portion of them affect humans. They affect us in our aquaculture attempts and in economics, but in biodiversity, they're also very important.
That's an interesting take naman, no, Doc. And yun po, sige po. May question, another question po from our Q&A chat box po. Sabi po, Doc JC, ask ko lang po if namatay na po ba yung host or madadamay na po ba yung parasite o lilipat lang ulit sa ibang host?
That depends on the specific taxon. So for example, if it has a direct life cycle, from one to another, kuwari na-consume, pwede yun. But hindi siya automatic eh. So with parasites that have very complicated or indirect life cycles, like yung mga pinag-aaralan ni Dr. Karin and ni Doc Gakad, they require intermediate hosts within a specific form. So if the final host were to partake of the parasite in a different form other than the next step, it won't actually be successful. So you have to wonder about that. So required sila. For example, trematodes. Digenetic trematodes somewhat require a mandatory gastropod or mollusk intermediate host. Diba? So it's never always the case. So when we talk about parasites, we have to specify which taxon we're talking about. So kasi iba-iba siya. But that's a good question. That's a good thing to think about.
Yeah, actually, Doc, I agree with that too. And regarding din po doon sa presentation ninyo, may another question on yung sa model host range po, is it an application? Clarification lang po kasi medyo mahina kasi yung internet daw nila.
Okay. Yun sa model host range, it can be applied for us to find possibilities. For me, yun lang talaga yun. You look into possibilities and see kung if it's actually possible. Kagaya na sa amin, I just looked into that if BIA can be a good host for the invasive fish tapeworm. And we were actually able to find that on a separate study na it was talaga. Diba? So, use it as a means for you to direct your attention. So for example, pwede mong baliktarin. Ano ba yung mga isda na meron ka sa area ninyo? Yun yung ipasok mo doon sa model. Tapos, ang gawin mo, pabaliktad. Check out all of the possible parasites that might be affecting the fish species in your own region. Diba? So, that's one way for you to do it. Pabaliktad naman siya.
Ayan, sige po. And to cap this discussion, Doc. Last question po from our participant here in Zoom. Can the use of a naturally occurring parasite as a biological tag in stock identification of marine fish be also used as a reference for fish history and identification for its endemic area?
Actually, that's possible po. That's possible. And people are actually doing that now. But it's very difficult. But if you're able to expound on that, it's a very powerful tool. Kasi para magawa mo yon, you actually need to be able to delineate the entire life cycle of the parasite itself. From kung may intermediate host man to the final host, diba? And you should have been able to obtain all of the parasite, that parasite in all of its stages within a particular region or area to be able to confirm. But once you're able to do that, you can use that now as a biological tag. Pero for me, that process, each step of those is a different study. So that involves at least 3 or 4 different studies before magawa mo siya. So pwede mo na yung pang masters, pang bachelor's degree mo, pang masters mo, pang PhD mo. Kompleto na yun. So by the end na yun, pwede mo na siya magamit.
Nice one. Kahit isang lang yun, Doc, no?
Isang parasite lang, di ba?
Talaga. Sige po. Ayan, sige po. Maraming salamat po, Doc JC, for that very fruitful po na discussion natin sa inyong presentation. And thank you po for giving us time na maka-attend po dito sa ating FishKwela activity.
Maraming salamat, Lea. Thank you very much. And thank you very much to the NFRDI. Maraming salamat.
Yes po. Ayan. So that was Doc JC po from University of Rizal System, our fourth speaker for this FishKwela activity. We will now proceed po to our next speaker. Ayan, ipapakilala ko sa inyo. So our next speaker, our last speaker actually, is currently taking his Doctor of Philosophy in Tropical Medicine and Parasitology at the Seoul National University College of Medicine, Seoul, South Korea. So he has earned his Master of Science in Biology at the De La Salle University, Manila, and his master class in Teaching Research at the same university. So he has published various studies, and some of his publications are the following: the novel assembled mitogenome analysis of Trichuris trichiura from Korean individuals using nanopore-based long-read sequencing technology, interaction of host, parasite, and environment laboratory, ocular microbiome analysis, of Stevens-Johnson syndrome patients in Korea Then soil-transmitted helminth egg contamination from soil of indigenous communities in selected barangays in Tigaon, Camarines Sur, Philippines. Then analysis of Trichuris trichiura whole genome using nanopore sequencing. Fatal granulomatous amoebic encephalitis caused by co-infection of Balamuthia mandrillaris and Acanthamoeba species, a case report, and growth analysis of Chlorella vulgaris in varied TMRL media and light exposures. So with that po, he is also a skilled bioinformatics analyst on the genome assembly and SNPs detection, recombinant protein production using molecular biology techniques, spider rearing for pest management and biological control. He's also Linux and R programming proficient. Ayan. He's also skilled microalgae upscaler and maintenance for biofeed and biofuel production, graphic designs, and he's also a computer software and hardware setup NC2 holder. So with that po, without further ado, allow me to welcome Mr. James Owen de la Luna.
Hi, good afternoon, everyone. My name is James Owen de la Luna. I will be presenting and sharing my research and expertise about parasite diagnostic. But first of all, I would like to thank Dr. Gakad and Dr. Muji for this opportunity and the rest of the team for organizing this event. So today I will be sharing our research with the title From Sequence to Clinical Insight, the Role of Mitochondrial Genomics in Whipworm Infection Management. So I was also asked to emphasize more on the molecular diagnostic techniques. So I will be sharing more about that later. Right. So allow me first to welcome you into the interesting world of parasite diagnostics and explore how the molecular techniques are changing the way we detect and understand these fascinating organisms. The parasites and their hosts, they have evolved together over thousands of years, with each adapting to survive and outsmart each other. And by using new diagnostic tools, especially molecular methods, we can better identify these parasites. And understanding how they work and even finding their weak spots. So this knowledge helps us develop better treatments and keep an eye on changes that could lead to drug resistance or new disease threats. And here's the exciting part. These techniques, they're not limited to parasites alone. So molecular diagnostics, they can offer insights across all fields of biology. So whether you're studying bacteria, plants, animals, or any other organism, you can make use of these techniques. So the methods that I will be sharing with you today, these are powerful tools that can help researchers like you and me to understand and analyze your subjects with great precision. So whether you're here for parasitology or just looking to apply new methods in your own field, there's something valuable that you can take away from this presentation. So let's get started. Right. Do you remember the last time you took a deworming tablet? All right. So if it's within the last 6 months, can you please type one in the chat room, the chat box? So I'm pretty sure that many of us here have likely not been dewormed for over a year now. Am I right? Okay. So how confident are we that we're not harboring or we're not carrying any of the parasites that the previous researchers or presenters have shown us? It's quite scary, right? We might have them, we might not. So how can we check? How can we check for these hidden infections and what methods can we use to diagnose them? So today I will walk you through those fundamental techniques for detecting parasites, both through morphological examination, where we look at their physical traits under a microscope, and also through molecular methods, which allows us to detect their unique genetic markers. So together, these approaches, right, so these approaches provide powerful tools for identifying parasitic infections accurately and effectively, and even when they're not visible to the naked eye. So basically, we have here most common parasite diagnostic examination. So we collect blood, fecal sample, and urine, examine them for the presence of parasites. And in my case, what I do in our lab in South Korea, since we're connected to Seoul National University Hospital, I mostly do molecular examination. So several hospitals sends us some samples, patient samples, if they cannot identify the infection, and we do molecular testing. So in this bench aid for diagnostic of intestinal parasite published by WHO, this is basically our Bible for parasite diagnostics. So inside this manual, it contains some techniques that you can use depending on what sample you're processing. And it also contains photos of references for you to check whether this egg is from this kind of worm or which I'd also like to share since we are in the morphological diagnostic step here. There are a lot of database that you can access, such as Swish TPH. They have this virtual microscope that contains database of microphotographs of eggs. So maybe you can access this one if you're also doing parasitology research. All right. So let me emphasize the two main categories of parasite diagnostics. So basically we have morphological diagnostics and molecular diagnostics. So both of them, they have some advantage and disadvantages. So for morphological, it's cost effective. You just have to have your microscope and your samples, of course. But the disadvantage is that it requires certain level of expertise to minimize error and make sure that you're actually giving your patients the right results. And that can be eliminated, the error, or can be lessened with the use of molecular diagnostics, which is highly sensitive and specific and can be automated. So even if you're not an expert, you can make use of standardized protocols that are already automated in the machine. And the only disadvantage is that it's quite expensive, especially the reagents and of course the machine. And you cannot bring them in the field, most of the machines. All right. So before we proceed into the workflow of molecular diagnostics, I want to have a little bit of review about our knowledge on genes, or particularly genomics. So what is genomics and why use mitochondrial genome? So a genome is a complete set of an organism's DNA. So they determine the DNA contains the gene, and then the gene determines the trait because they produce the protein, right? DNA to RNA protein. Okay. So inside the cell, every living organism, they have the cell. You have the nucleus, and inside the nucleus, you have the chromosome. The chromosome is basically a condensed form of DNA. So inside the cell, you can have nuclear genome or mitochondrial genome. This is the nuclear genome, the ones found in the nucleus. While mitochondrial genome, these are found in one of the organelles of the cell, specifically the mitochondria. So the question is, why do we use mitochondrial genome most of the time in parasite diagnostics? Right. So the use of mitochondrial genome instead of nuclear genome is quite useful. Why? Because they have a higher mutation rate, meaning to say that they have clearer genetic signatures for identifying species and tracking their evolutionary history. Later on, I'm going to talk more about the evolutionary analysis. So this is especially useful for diagnostics because it allows us to detect and differentiate organism even when only tiny amounts of DNA are present. And since there are more mitochondria inside the cell, it's quite logical to make use of mitochondrial genome. But still, depending on the kind of question that you want to answer. All right. So let's proceed. So here's a molecular diagnostic workflow showing us after collecting your sample, what do you do next? So you do morphological identification using your microscope, dichotomous key, and then the reference guide. And you also extract DNA and do PCR and DNA sequencing. And once you have your sequence information, you can upload it in the database and compare and check with the previous references. And it will tell you the identity of the organism that you're trying to check. Or you can also just extract the DNA, do sequencing, and upload. And once you have your identity, you can do further analysis. Right. So basically, PCR, it detects the parasite present in your sample by making many copies of your gene, okay, that identifies the parasite. While for sequencing, it is performed to analyze the genetic information further, whether you want to check the identity, if it's a novel species, or you just want to check for presence of mutation, specifically drug resistance. So those are the things that you can do. So here's another slide showing or emphasizing the difference between PCR and sequencing in the molecular diagnostic workflow. So after collecting sample, you extract the DNA, you do PCR to check if your sample is positive or negative for the infection or the parasite in this case. And once it is positive, you collect the bands. So these are the bands that tells you that your sample is positive. You collect it, extract from the gel, you do sequencing. So in the sequencing process, which I will show later, the breakdown of how it's performed, basically it will give you an output of DNA sequences like this. All right. So once you have your DNA sequences, you can align it with a reference from the database. And now you can do further analysis such as multiple sequence alignment to detect variant and also do some evolutionary analysis or phylogenetic analysis. We usually hear the word PCR, especially during the pandemic. Oh, have you done your PCR test? So what is PCR? So it's actually a technique that is used to amplify or make many copies, millions of copies of your desired DNA to confirm the presence or absence of an infection from a sample. Okay, so here's the steps. So basically you subject your sample to series of different temperatures wherein the DNA polymerase will attach the nucleotides to your template, and then it will per cycle it will produce more copies of your target DNA sequence. So after 35 to 40 cycle, you will now have millions of copies that will be used to confirm that indeed the infection is present in your sample. So here are some questions that you can use to answer some research question by doing PCR and sequencing. Okay. So for PCR, it answers the question whether the parasite is present in your sample and how much of these parasites are present, especially if you're doing qPCR. And for sequencing, if you want to check the variations or mutations among this species, you can do further sequencing. Or let's say you want to do evolutionary relationship analysis or check if the infection is novel, just like with the coronavirus. How did they know that it's novel? It's a new species. Of course, they have to compare it with the old species, right? So how do you compare it? You have to have a DNA sequence from this new species. So in short, PCR is for detection, identification, and quantification of parasite in your sample, while DNA is for further analysis, checking genetic information and providing you with evolutionary context and discovering variations or even new species. Right. So here are a list of markers that are commonly used in parasite diagnostic. Actually, there are more. And depending on the organism that you're trying to study, they have their own specific markers used, but this is the most common, right? So we have the COX-1 or the cytochrome C oxidase. This is part of the mitochondrial genome. I've already mentioned the mitochondrial and nuclear genome, right? So COX-1 is a portion of mitochondrial DNA or mitochondrial genome. And ITS, this is part of a nuclear genome. So they have a specific questions that they answer. So if you want to check for specific species, let's say if you want to differentiate or confirm, oh, this is from a pig sample and this is from a human sample. So you can make use of COX-1. Or if you want to check, oh, this fish is different from this one because the COX-1 gene marker result is like this and like that. So you can make use of COX-1. And also, if you want to answer specific questions such as, does this infection already develop resistance to this specific drug? So you can make use of gene markers such as beta tubulin gene. Beta tubulin is actually a gene that codes for protein that are responsible for the structure of the cell, cytoskeleton structures. Right. So these are gene markers that you can explore further in order to check which ones you can amplify to tell you more about your infection. All right. Now let's move on to DNA sequencing workflow. So here we can see a sample with several types of microorganisms. But if you just want to study this one, so first you have to do DNA extraction and then remove the DNA from the contaminant. So you remove the unwanted genetic materials and then you prepare the library. When we say library, basically these are DNA sequences that are extracted and then you attach some adapter. The adapters are proteins that will guide your DNA to pass through the sequencing platform. So in this case, we are using third generation sequencing technology, Oxford Nanopore. So actually there are three generation of sequencing technology. First generation, Sanger sequencing. Second generation, short read sequencing, it's like the Illumina. And third generation sequencing, long read sequencing. This one, Oxford Nanopore or PacBio. They just differ in the amount of nucleotides that they can produce or sequence. So for this one, it's capable of producing up to 10,000 base pair. But for the short reads, just up to 500 to 1,000. So long sequence. All right, so for this one, how do they produce this sequence of letters? So you have your extracted DNA sequences with the adapter. The adapter will guide this DNA to pass through this nano-sized pores, as you can see here on the screen. And every time they pass through these pores, they measure the electrical conductivity. So each nucleotide, say for example, cytosine, it has a specific signature, electrical signature that is different from guanine and different from thymine and adenine. So those difference in conductivity is measured and corresponds to specific nucleotides. So that's how they produce their sequence of letters. That's now your sequence of DNA. Right. So in our lab, we make use of that nanopore sequencer. So once you have your DNA sequence, what do you do next? So you align it with other sequences from other organisms or same organism to check which one is closer to which. So you do sequence alignment and phylogenetic analysis. So using parasite sequence for phylogenetic analysis is basically like making a parasite's family tree. So you're looking at who's closer to who. And you're making use of the genetic information to draw the family tree. It looks like a tree because it has a root it has branches, and these are the leaves of this organism. So you're trying to check what is the most common ancestor, who is closer to who in terms of their genetic information. So by comparing the DNA sequences from different parasites, so it gives scientists an idea on how closely related they are, and they reveal also the evolutionary paths that they've taken. And it helps us understand how certain parasites have evolved over time and how they are related to each other, and even how they may have adapted to different hosts or environment. So those are information that you can get by doing phylogenetic analysis. So in a way, it's using genetic maps to trace parasite ancestry and help us better understand place within the larger network of life. All right, so now that we're on the same page when it comes to parasite diagnostics, you're already familiar with the basic molecular techniques and the concept behind it. Let me share with you our research that was conducted a couple of years ago. So we collected samples from patients. We did some surgical extraction of parasite because the patient is complaining from abdominal pain, and patient have rectal prolapse and growth retardation, which are the effects of having parasite infection. So in this case, we have identified it to be a whipworm, human whipworm. So it's part of the long list of neglected tropical disease. NTDs, or neglected tropical diseases, are infectious disease in tropical countries, basically. And one of them is soil-transmitted helminths, STH. By the name itself, part of their life cycle, they are transmitted into the soil. Once an infected person defecates, it's lack of hygiene and sanitation, and then the cycle continues. You get infected when you accidentally eat an embryonated egg. So maybe you didn't wash your hands properly, or you didn't wash the vegetable that you eat, or you ate some meat that are contaminated with this parasite. Right. So in South Korea, the prevalence of this infection is very low because they have very good healthcare system. But the question is, where do this infection came from? Even now, they cannot eliminate it. Is it being transmitted from other country? Do they develop already some drug resistance? Why are these worms still persistent? So those are the questions that we try to answer. So this is what a human whipworm look like. Quite scary. So the head is actually on the small end of this worm, not this one. This is the tail. Okay, the head is the small part. And they are smaller than a matchstick, but they can produce up to 16,000 eggs per day per worm. All right, so it can produce a lot of eggs. So they are usually found in the small intestine because that's where nutrients are absorbed. So they're just waiting there. All right, so in South Korea, as I mentioned, during 1971, the infection rate of whipworm is quite high, staggering 65%. But as the year goes by, the infection goes down. But even till now, it's still there. So last survey was 2012, so 0.4%, quite low, right? So what does this data tell us? So it's difficult to eliminate this infection, and we need a more sensitive diagnostic tool if you want to detect further transmission of this infection. So in the database, there are some genomic information, mitochondrial genome of Trichuris trichura. So we have some reference from China, from Japan, but it's not verified yet, and from South American countries. But in South Korea, they don't have genomic information about whipworm parasite extracted from humans. So in this study, we tried to confirm what is the molecular identity of the worm extracted from our patient. And also, we would like to contribute in the database. We would like to produce the first mitochondrial genome of whipworm extracted from Korean patient and using the latest sequencing technology. And using this insight, we would like to provide some guidelines to healthcare professionals on how to manage whipworm infection. So all of these objectives are aligned with sustainable development goals. So when it comes to the methodology, so same thing with what I discussed earlier. So we extract the DNA from patient sample. We do PCR and DNA sequencing. We assembled our sequence, and then we were able to produce mitochondrial genome. So it has a lot of steps, bioinformatics steps here during the assembly annotation. So basically, we did quality control. We want to make sure that the sequence we produce is actually the sequence of the whipworm. So after doing quality control, we submitted in National Center for Biotechnology Information Database, or NCBI. And after passing their protocols for quality control, we finally have our mitochondrial genome of parasite from a Korean patient. So this is the first mitochondrial genome of whipworms extracted from a Korean patient. So first data for Korea. Ironically, it was assembled by a Filipino guy. That's me. So my dream is to also produce a mitochondrial genome extracted from a Filipino patient. So if you know someone who have a whipworm infection, kindly connect that person to me so we can extract the worm and then also create a mitochondrial genome from a Filipino patient. All right. So we not only produce one, we were able to produce 3 mitochondrial genome. So now that we have this data, so it contains several portion of the mitochondrial genome here. So this is the COX-1 region. We also map it or align it with other sequences from the database. We have sequence here from Japan and sequence from Uganda and from China. So we also compare them in terms of their properties. And we are happy to share with you that our sequence have better quality in terms of 80 repeats. It has longer 80 repeats region. So in here, what does it mean? So it means that we have produced a better quality of mitochondrial genome because those regions that were not previously reported, we were able to cover and assemble them. So since it has a longer 80 repeat region, it can now be used as a genetic marker for resolving intraspecific infection, especially if you have some mixed infection. They can use it to answer how many mothers are there, how many mother worms are there in the patient. All right, so for the phylogenetic analysis, so here we're showing the phylogenetic relationship between samples from different countries, as well as samples of whipworm from different hosts. So we have here whipworm from pig, whipworm from monkey, and whipworm from humans. So in here, we can find our samples from Korea. So 2 out of 3 of our sample is closely related with the ones from China. And 1 out of 3 is related to the ones from South America and Japan. And it's even clustered with 1 monkey sequence. So quite interesting. So this one, sequence 2 and 3, they're closely related with China. So we asked the patient, do you have some travel history to China? So lo and behold, they actually frequently travel to China. So we are now suspecting that this infection was transmitted to Korea. But we have to do more analysis on that. All right. So one good thing about this tree is that there's still no zoonotic crossover, meaning to say the ones that infect humans are still infecting humans and not monkey. And then the ones that infects monkey are still not infecting humans. They're not cross-contaminating each other. And the ones that infects the pig, they're not yet contaminating or infecting humans. Otherwise, it would be a different story and quite scary if the ones that can infect the pigs can now infect humans, right? Right. Okay. So our study highlights the significance of having a mitochondrial genome data to give us some overview of how they are transmitted, how they're closely related with each other. So in this study, we were able to produce a high quality genome with an enhanced genomic resolution that can potentially be used for diagnostics. Right. So what do we recommend now using this data? So we are recommending to the public health officials to do some periodic deworming program to further lower down the infection rate and also strengthen cross-border surveillance to prevent the transmission of this infection. So what's next? So currently we're doing research on anti-helminthic drug resistance of whipworms in the Philippines. So we're collecting samples from Filipino patients and check if there's already a drug resistance to anti-helminthic drugs. Since we've already done a couple of decades of national deworming, but until now, there are children that are still infected with whipworms. What is happening? Maybe there's already some drug resistance. So we're on this stage already. So thank you very much for listening. And that's all for my presentation. And if you want to collaborate with us, please email or you can send us a message. And thank you very much for your time.
Ayan, thank you po, Doc Joanne. Doctor na po. And future doctor. Ayan, hi po, sir.
All right, good afternoon, everyone. Can you guys
Ayan.
rinig po ba ako? Can I see some thumbs
Okay.
up? If you guys
Sige
can
po,
hear-
good afternoon. Sorry po.
Can I see some thumbs up if you guys can hear me? All right.
Yes, yes. Opo.
The response of the participants.
Ayan, sige po. We'll start the discussion po with some questions po on our chat box. Ano. From YouTube Live po ito. What are the difference between nuclear genome and mitochondrial genome? Can they produce different results daw po ba?
Yes, that's a good question. So depending on the kind of research question that you would like to answer, you have to make sure that you're amplifying or targeting the right marker because it answers a different question. So they have a very different results and they also have their own specificity and depending on the organism. And if you do a lot of article review, they will also report like the golden standard when it comes to what kind of gene markers to use. But in our case, we prefer to use mitochondrial genome.
And sige po. And another question from our participant. So given that Trichuris tricura is an example of soil transmitted helminth, do you think that the conditions of the soil daw po has an impact? And can PCR and other molecular techniques be done on soil samples?
Yes, that's a good question as well. When I was doing my master's thesis, I actually did soil collection and we also measured the physicochemical parameters that affects the eggs and how they were able to survive those kind of conditions. So yes, the environmental factors affect the egg and the same thing with what Dr. Gakad have presented. You can also make use of environmental samples to detect the presence of parasites. So yes to both questions.
And sige po. Moving on to the next question. Good afternoon, sir. I just want to clarify what is the important role of mitochondrial genome of parasites in their biology and evolution?
Right. So since they are quite different from each other, you have nuclear genome and mitochondrial genome. They play a very different role when it comes to production of proteins and gene regulation processes. So they have their own specific roles. And based on the literatures, there are also instances where they coordinate with each other to maintain some cellular processes.
Interesting. And another question from our participant. Given the migration theory, is it applicable here in the Philippines locally? I think this was for the infection na nireport nyo kanina doon sa ano po.
Yes,
Yes.
definitely. And most first world countries, the reason why they send more funding to tropical countries, especially parasitology research, is that they don't want the infection to go to them. So they're trying to resolve it on our level. I think most scientists here can agree that, yeah, they receive funding so that we can solve the problem on our level so that we don't have to bring the infection to other countries. So locally, it also happens. We are already familiar with the pandemic events, so we are familiar with disease or infection transmission.
All right, sige po. That's for the migration theory question, and another question from our chat box po. Doc, bakit po minsan daw pag nagpurga wala namang nakikita na parasite sa poops?
So good question. So not all antihelmintic drugs will flush out the worms. No, some of them they're not flushed out as an entire worm. There are drugs that just break down the worms while they are still in your stomach. So you don't usually notice them when they're passed out through the feces. No, and some of them they just paralyze the worms and then wait for some time before they are flushed out.
So hindi porket wala kang nakita, hindi na totoo.
Exactly. Yes.
Sige po. Another
And
question po.
also, yeah, to add, hindi porket nakita mong may lumabas na isa, naubos na lahat. That's why sometimes we also recommend patients to do like a 3-day deworming session just to make sure. And also the proper dosage is important.
Ayan, sige po. And another question from our YouTube live participant po. How much or how different are molecular analysis studies po? Where in the Philippines can it be conducted?
Many universities, especially those who have parasitology units, they conduct molecular analysis as well. So when I was still working in UP Manila, College of Public Health, we also do molecular detection of parasites, but we're mostly focused on schistosomiasis because it's the number one deadliest neglected tropical disease. Previously, China was number one in terms of the prevalence, but they were able to resolve the problem. And now, guess who's the number one? Philippines is the number one. Sadly, number one in terms of the highest infection rate of schistosomiasis. So hopefully through this kind of platform, we encourage more students to do medical research, parasitology research, because you guys are the next generation of scientists that will resolve potentially this problem. So thank you for being here.
Ayan, sige po. And another question po, this is from YouTube also. Can you tell us any more info or information about the antihelmintic resistance?
All right, so as we know, these organisms are also smart. They don't have as big brains as we have, but they're quite smart. They can adapt to adverse environment as well as the drugs that we use to kill them. So they can mutate and they can develop resistance, especially if you're not taking the drugs as prescribed. Same thing with bacteria, no? They can develop resistance as well. And currently, we are already suspecting that there are resistance cases, but we, of course, have to confirm through laboratory and molecular testing. There are cases in public schools wherein we only observe whipworm infections, but the roundworm infections and tapeworm infections were eliminated already. So, bakit whipworm lang natira? Usually, when you deal with soil-transmitted helminths, trinity yan. We have the roundworm, the whipworm, and then the hookworm. Laging magkakasama yan. Best, mga mag-BFF yung tatlo. Pero ngayon, bakit whipworm lang yung natira? Baka yung whipworm resistant na. Mas malakas na siya doon sa dalawa. So those kind of questions we're trying to resolve and answer.
Very interesting take from our participant. And another question po from our Q&A box, kailangan po bang matanggal lahat ng lahat ang mga parasites sa katawan ng tao?
Alright, that's a tricky question, ano?
Yeah.
Parasites, they live in our body, and I'm not pretty sure if we can survive the evolutionary process kung walang parasites that are interacting in our body. They sound like they're the bad guys, but actually they also have advantage. As I mentioned in the introduction part of my presentation, it helped us evolve
Yep.
because of constant banter. It's constantly bombarding you with infection, and your body is trying to evolve and develop to combat that infection. It makes you stronger. Sabi nga, what doesn't kill you makes you stronger. And the parasites are not after killing you because they live inside you. So it actually makes you stronger. Something like that.
Ayan, sige po. That was fun, Doc. Thank you very much po, Sir Jowen, for that fruitful discussion po. And thank you for giving time. Kasi alam din namin na medyo busy din kayo sa work, no? Dyan sa South Korea pa rin ba kayo, sir?
Nandito ako sa Pinas, but I'll be going back to Korea next year.
Okay. Sige po. Ayan, maraming salamat po, Sir Jowen. And yan, so far, ang ating mga presenters po for this FishKwela activity. Yan, maraming salamat. And for further questions po na hindi namin na-raise sa atin po mga participants, we will be forwarding them po your queries para sila po mismo ang sasagot sa inyo through email. So ayan, maraming salamat po sa atin po mga resource speakers, sa active participation ng mga participants natin. And let us go to the next part of this program. Ayan, so para po ma-assess namin kung gaano kayo natuto sa atin pong FishKwela. Ayan, kindly scan the QR code or go to bit.ly/post-paramap para po sa ating post test for this activity. Ayan, sige po. Ayan, link. Punta po kayo sa inyo pong browser, bit.ly/post-paramap. Ayan, sige po, hayaan niyo lang po, nakaano naman po siya, naka-open lang naman po siya in case po na hindi niyo pa po ngayon siya ma-access. So you can, like access po it later sa atin pong, after po nitong ating FishKwela activity. Ayan, sige po, yes, ayan po, we would also like to hear from you. Please evaluate po this activity by scanning the QR code on your screen or going to bit.ly/evaluate paramap. Ayan. Ayan, yes po, hindi niyo po talaga siya ma-view as ano po, kami lang po siya makaka-edit niyan. Pero naka-ano naman po siya, naka-public naman na po siya. So, try po accessing it later on. So, ayan, yun po para sa atin naman pong evaluation. And yes, reminder for our participants to please visit our bit.ly/attend paramap 11:30 na attendance form link. And po naka-share po sa screen natin. And then huwag po ninyong kalimutan talaga na mag-attendance kasi this will be the basis of the issuance po of e-certificate as well as the CPD points for this activity. So, participants who will complete the requirements, ayan po, i-reiterate lang namin, participants who will complete the requirements, the following requirements po, the attendance form, the pre-test. The pretest po is appended doon sa registration form. So kung hindi po kayo naka-register, wala po talaga siguro kayo ma-access na pretest kasi na po namin siya, na-i-administer na namin siya during registration. So pretest, post-test, as well as yung evaluation po sana, yan po yung magiging basis namin para i-issue yung sa PRC. Okay. For new passers po na wala pang PRC ID, hindi po kami sure kung ma-i-issue sa inyo if ever. Pero pwede nyo po siya i-apply actually sa PRC Leris database. Ayan, kailangan nyo po yata mag-sign up for Leris. And kailangan nyo po siyang i-upload doon yung i-issue namin e-certificate. So makukuha nyo po yung karampatang CPD points. Yan. Sige po. Okay, sige. And we will now go to the awarding of certificate for our resource speakers. Yan. Yan, sige po. Next po, sir. So the certificate po, the first certificate, the Department of Agriculture, National Fisheries Research and Development Institute awards this certificate of appreciation to Dr. Karin Chuchida in recognition of her valuable expertise and knowledge shared as resource speaker during the FishKwela webinar on paramapping, morphological, and molecular techniques in parasitology held on October 30, 2024 via Zoom. Given this 30th day of October 2024, signed by our Executive Director, Dr. Lilian C. Garcia. And the same certificate will be issued to Dr. Janelle Laura J. Gahad. Yes. The same certificate will also be given to Dr. Arvinjet B. Marcaida, to Dr. Jonathan Carlo A. Briones, and to Dr. Mr. James Owen C. De La Luna. Ayan. Congratulations po to our speakers. And thank you very much po for giving time na ma-share po ang inyong knowledge and expertise dito po sa aming FishKwela platform. Okay. Ayan, sige po. And with that po, I am now giving the floor to our training specialist 3 / our training section chief, Ma'am Norby Gatdula, for her closing remarks. Hi, Ma'am Norby.
Hello, Lay. Nagkakaroon tayo ng mga problems ba? The show must go on. So, ladies and gentlemen, mabuhay. As we draw event afloat, I want to thank each of you for your enthusiastic participation and engagement throughout today's enlightening session. All this wouldn't be possible without you, of course, the participants. From our deep dive into trematodes and nematodes with Dr. Karin Chuchida, to exploring cutting-edge parasitology techniques with Dr. Janelle Laura Gahad, with expanded our horizons and enriched our understanding of this fascinating subject. Dr. Alvin um, Marcaida shed light on the phylogeography of rabies species, parasitic nematodes, while Dr. Jonathan Carlo Briones emphasized the importance of big data in fish biology and fisheries. Our journey concluded with Mr. James Owen um, offering of invaluable insights into mitochondrial genomics and reform infection management. And we knew more about the reform now. And we appreciate your sincere effort in making every participant understand the value of paramapping. Yes. So, kahit ako po, medyo busy po talaga ngayon. I am hooked in this lecture para matutunan ko rin naman kung ano yung mga topics na ito. And today's discussions were testament to the vibrant and collaborative spirit of everyone. Let's carry forward the knowledge and connections we've made here to foster further research, collaboration, and innovation. Yan. So, natutuwa ako dun sa mga resource speakers po natin na talagang who are inviting us to collaborate with them. Of course, let me thank the support of our Executive Director, Dr. Lilian Garcia, and our Division Chief in Training Division, Dr. Mujiki Santos. And of course, my team behind the organization and execution of this event is Leia De La Cruz and Joey Cater, who, in spite of being under the weather right now, actually under the weather po silang dalawa, but they have exhibited their professionalism. Of course, thanks then sa same thing for the for really contributing their time to this. They deserve all the credit for making this happen. Don't you think they deserve a round of applause? Okay. So, um, our team actually always strives to give you quality training and add value to it by applying for continuing professional development CPD points to PRC to support fisheries professional. Our our application for CPD points is entirely dependent on the approval of PRC. And we announce our activities ahead of time to give everyone ample time to register for the event. Even before we apply the CPD points or kasabay din po yan ang application namin sa CPD points. And of course, we can only announce the CPD credit once we get approval from PRC. So that is why later na lang po namin nasabi that this is CPD bonus will be accredited, will be credited in this training. So again, thank you to our resources speakers. Thank you for giving us your time. And thank you everyone. And we hope to see you in one of our future events. God bless everyone.
Ayan, thank you very much po, Ma'am Norby. Ayan, maraming salamat po sa inyo. And thank you very much din po sa ating mga participants po na naging patient po sa atin pong mga pagsisend no, ng mga links na kailangan para masagutan yung mga post-test, pre-test, post-test, evaluation form, ayan. So, ayun po, that ends our FishKwela activity for today. Ayan, maraming salamat po for your usual support. And salamat din po sa sa lahat po ng mga tumulong para mabuo po itong FishKwela activity nito. Thank you very much din po. Actually, magpapasalamat na din ako dito sa sa IMTS, ayan, headed by Sir Reggie Pistaño. Thank you very much po, Sir Reggie, and Sir Jolly for assisting us para maging successful itong activity na ito. Ayan. So, ayun po, thank you very much po sa atin po mga resource speakers. Yan na naging patient sa mga questions na ni-raise po natin kanina and for giving us time despite of their busy schedules. So with that, we will now be ending our FishKwela activity. This has been Lay, your MC for this FishKwela session. Saying maraming salamat po and ingat po sa lahat ng mga uuwi, lalo na this weekend. Ayan, maraming salamat po. Bye!